it's not connected. So, just a second. Is it working? Yeah. It's taking more time. So actually, till now it was not connected. Just now it connected. Just now only. So now it will come. Yeah, now it's connected.
Echoes.
The last operation disconnected the
Because the signal is not going from here. Yeah. For that, HDMI is connected, so you can Sorry? No, don't worry about me cheating. Yeah. I slept the whole night.
This was
Yeah, this is connected.
This is connected.
Yeah.
Yeah, say like connection.
Okay.
Fine. This is all.
Is there anything?
This is not on. Power
This is power is coming.
Should I put my laptop here? Is that easier? But then the sound there,
Yeah.
it's connected there to the. No, I have that movie. I'm not going to change it. I'm going to run it from my laptop. Otherwise, things change.
beamer on? The beamer is on? It was on, but it doesn't get a signal. Can you, I think you put the beamer on first, Kumar? You put the beamer on first and then we put the input, might work. Wait a second now, but not yet. When it's on, when it's asking for input,
Put this projector. Yeah, it's just for it. Remote.
Directly to what? This one.
is that connected?
This cable is connected to
Give it
this laptop now.
We're not connected.
Yeah, so first put the beamer on and then we connect. Yeah.
Sir, which
He is going to use this.
what is which one is the lead of the projector?
That's this one, you know.
No, sir, not remote.
I'm gonna switch up the
Board of
I don't know that.
Okay, give it here. He knows it.
Yeah, then this one goes here. Yeah, so it goes like that. Screen, and then it goes here.
ये कनेक्ट करना है
अच्छा लैपटॉप से
हाँ
आपको ये स्क्रीन दिखी वो स्क्रीन दिखी
So I think for tomorrow, if it works, let's leave it as it is. Right. Yeah.
Always an issue.
Hey, morning. Yeah.
But the beamer is
So the beamer is.
yeah, yes.
Wait, just don't wait yet. No.
It's coming.
Yeah.
still not from sound projector will take some time
HDMI would also work, right?
Yes.
Yep.
You should try to connect it.
Use this one, just take it off. Yep, take it out. This has an HDMI.
will start today's lectures. And of course, the program is already shared with all of you. And follow the lectures and practicals today. Thank you.
Okay, good morning. Let's get started today. First of all, I want to give my compliments to the team and the group yesterday, because it was a long first day, correct? With the inauguration and the lectures, and then both the computer practicals and the wet lab. But so I think today we will have a bit more time to to go through, because it's in the afternoon. It's primarily computer practicals, right? So we will have time to go through that, and we started 1 hour earlier. Well, okay, a bit earlier. So the lecture today. So yesterday, Victor gave a talk about microbial diversity, right, and the dark matter, microbes that we don't know yet or we cannot culture. And today we will go more into microbes that are associated with plants. Good. So let's first start with a definition. I saw also in the entry exam yesterday that there was a question on, I think, a definition, or the first person who coined a definition of the microbiomes. If I'm correct, I think that was Joshua Lederberg, which was one of the Nobel laureates, bacterial geneticist. And I think what is important to emphasize here, that the definition of the microbiome is the totality of microbes, but also their genomes and their interactions in a particular environment. So it's more than a microbial community as such, right? And it's interesting because if you look at the genetic information that is present in microbes associated with plants, but also with humans, with insects, there are so many more genes and functional potential there than in this case, the human genome or the plant genome, that they also call this the second genome of eukaryotes, because it basically expands the genomic and functional potential of the eukaryotes. Victor also mentioned the human microbiome project, which was initiated already about 15 years ago, and which actually also propelled the field of microbiome research. Yeah, we are in India now. I think, if there is an opportunity in the future when you come to the Netherlands, you fly into Amsterdam airport, and in Amsterdam there is a museum called Micropia. It's I think worldwide. I think there are a few more, but this is one of the nice examples of a museum which is fully dedicated to microbes and the potential of microbes in ecosystem functioning, and many other elements are actually described in that museum. So I'm just going to show you, it's sort of like if you go to the website of Micropia, you will also find this movie. So we're going to try to run it with the sound. I'm not sure how it's going to work out, but we'll give it a try.
I always thought I was alone. Apparently not. Seems I've got trillions of microbes to keep me company. Under my armpits, on my hands, under my feet, everywhere, in fact. And what's nice, they stick by me, which is actually a good thing, because microbes protect you. We've all got them, and we're constantly swapping them with each other. Our whole life revolves around microbes. They're in your food, in your tongue. The microbes can do hundreds of thousands of things. Meet your microbes in Microbiomes.
So this is actually, it's a super nice museum to go to. like I said, it describes, the tree of life. When you enter the museum, you show the tree of life. You see humans as a very small branch of the whole tree of life, like Victor showed yesterday. And there's many places. There's a section in the museum where you can smell microbes, so related to volatile compounds that they produce. There is also, a place where if you are, with your partner there, and you give each other a kiss, it will tell you how many microbes you actually exchanged. So there's all these little features there, which makes the museum super fun to go to. So that's okay. Let's go to the questions in microbiome research. The lecture is about plant microbiomes, but actually. So the lecture is about plant microbiomes, but the questions I'm addressing here, those are actually questions. So if you're interested in insect microbiomes, the same questions apply. So there are always three sort of like starter basic questions. So who is there, which relates to the taxonomic diversity. And also that today with the computer practicals, you will go into determining the taxonomic diversity of the microbiome. Where are they located? There's not so much research done on that, but in terms of localization, you can do a lot of methods like microscopy, but you can also do fluorescent in situ hybridization to localize specific members of the microbiome in a particular sample. And the field, if you look at the field, where the field is going is what are these microbes doing in the system. Again, coming back to the definition, what are their interactions in that particular environment and what are the underlying mechanisms. So you see that the field is moving from a stamp collection, and I mean that in the positive sense of the word, so describing, okay, this is the collection, but what is their function? And it's interesting, I always use this analogy, so I'm not sure if there are people here who collect stamps, but if there are, you know that sometimes, a single stamp has an enormous value. But sometimes stamps do not have a value unless they are part of a bigger collection of stamps, right? And look at it at microbiomes in the same way. So sometimes you have a particular microbe that has a big functional impact on the system that you are working in. And sometimes you need, consortia, or nowadays they call that synthetic communities, to actually give a particular, functional trait to the system. So keep the stamp collection in mind. Then several of the key questions that many microbiome studies address, and sometimes they don't address that. The first question actually seems super straightforward. So to what extent do microbiomes actually influence a particular function of that system? So if you're working on plants, what is the relative contribution of the microbiome to a particular phenotype of that plant, right? Or if you work on biogeochemical cycles, what is the contribution of the microbes to carbon cycling or nitrogen cycling? Many times this question is not addressed specifically. Usually there is sort of like an assumption that microbes must have, a function in that particular ecosystem. I will come back to that later when we talk about methods how to study microbiomes and the functionality of microbiomes. Then a very interesting question, and we will come back on that in tomorrow and Thursday's lectures on how do microbes assemble on eukaryotes? And how do eukaryotes basically recruit microbes for a particular function? We will talk about that. The third question, I think, is the most difficult one to answer. We are not there yet. Are there general patterns in the dynamics of microbiomes and also their functioning? Because I think once you know and understand the patterns in this dynamics, I think you can also engineer microbiomes to what you would like the microbes to do. And that's the last question. So how to engineer these microbiomes to improve growth and health, because that ultimately is the sort of like the point on the horizon. So when you study microbiomes, you need to have a systems approach to understand their functions. Okay, so what I'm going to do in the next few slides, which, so don't think that you ended up in the wrong course, but I'm going to talk about the human microbiome, and then at some point we're going to switch to the plants. These are pictures you can, so these all came from the Human Microbiome Project. You can see that, through age, your microbiome changes, but also during your lifetime, depending on, exposure to environmental conditions, or for example, to antibiotics or different foods, it will have an impact on your microbiome. And then, of course, also the genotype. So each of us, we have a microbiome that is similar, but you also have a microbiome that gives a signature to you, right? So, and I think Victor and also others will talk about the core and the accessory microbiome in that context. Okay, so, and then if you look at the two major topics in terms of functions that in microbiome studies always show up is health and disease and growth and development. And for the human microbiome, there's a lot of emphasis on how microbes impact, for example, on growth and especially on obesitas, right? That's a big theme within human microbiome research. And there's this famous experiment where they looked at sterile mice. And what they did is they transplanted microbiomes from lean mice to these sterile mice, and also microbiomes, gut microbiomes from obese mice, also to these sterile mice. And then they provide them with the same food source. And then you see that the microbiome from the obese mice, results again, in an obese mouse, right? That's that. So those are sort of like transplant studies where you transplant a whole microbiome from one organism to the other, and then you look for the phenotype. Of course, once you have that phenotype, you are especially interested in which microbes in that microbiome, are mostly responsible for that phenotype. And there's a paper here at the bottom. It refers to a bacterial genus, Akkermansia. And Akkermansia, that was one of the microbes that was found which controls diet-induced obesitas, right? So again, there are more microbes that contribute to that phenotype, but this is one of the first studies where they actually went from a transplant study to the identification of a specific microbe in that community that gives that particular phenotype. So, the last, I think, 5-6 years, there's also a lot of interest not only in our physical health, but also how microbiota contribute to our mental health. And it's super interesting. If you see on the slide, if you go from the microbiota from the gut all the way to the brain, they call that the gut-brain axis. There is an influence of the microbiome on stress, anxiety, mood, behavior, and the underlying chemistry there and mechanisms has to do with neurotransmitters. And vice versa, the brain can have an impact on the gut microbiota through also hormonal effects that you will have, which then changes the microbiota composition, which then in turn has an impact on the brain. So this is a picture that sort of like summarizes all this, and you can see it's quite elaborate. So associations of microbiomes with metabolic diseases here, there's a number of listed here. So obesitas I mentioned, but also, for example, NAFLD, that's a non-alcoholic fat liver disease. That's also, there is an association. And then here, the brain disorders, that's also listed here. Respiratory diseases, cystic fibrosis, I think that's a very good example. Skin diseases, the skin is the biggest organ of the humans, right? And there's a huge number of work done on skin microbiota and how you can improve that. And of course, there's a lot of work done on Crohn's disease, so the bowel diseases, and how the gut microbiota have an impact on that. Just to go a little bit more into detail, and again, you know, the references are here, and I will provide you with the slides as well. There's some super interesting literature on how specific, mostly bacterial species here, have an impact on specific hormones like oxytocin, which then transmits through the gut-brain axis into, you know, behavioral changes. So oxytocin levels go up, and also social behavior goes up. So many of these studies have been done in mice and have not been validated yet in humans, right? So keep that in mind. The other thing which you always have to keep in mind in these studies is cause or consequence. So are the microbes or microbial changes in the gut microbiota, are they the cause of, you know, basically the phenotype? Or is the phenotype of the disorder, does that have an impact on your microbiota composition, right? So, and that's, so there's a lot of studies that are been done to validate actually the importance of specific microbes. So gamma butyric acid here, that's a lactobacillus, also has an impact on your stress responsiveness and anxiety. And there's a number of effects of microbes on either the host producing or changing the neurotransmitter production, or the microbes producing specific neurotransmitters themselves. Again, I would advise, and have a look at this review paper, super interesting to read. So this is sort of like to summarize a summary slide of what is known from the human microbiome. So on the left, what is known, which gives sort of like a taxonomic picture here, the number of genes, the metagenomes that have been mapped, and then on the other side, what is still unknown. So the unknowns is actually, there's a lot of still hidden taxa, and there's also a lot of functional traits that are yet unknown. Excuse me. There's still a lot to learn. There's a number of genes there which are still not having a match in functional databases. Okay, let's move to soil and plant microbiomes. And again, for the picture that I've shown for humans, effects of age, host genotype, diet, and soil, in this case soil type, also applies to the plant microbiome. Going from seed germination here in the bulk soil, all the way up to a first seedling. And then again, you can put all the terminology there. The spermosphere, so that's the area surrounding a germinating seed. Then we have, of course, the rhizosphere, the rhizoplane, the endosphere, and then when you go to the above ground parts, it's called the phyllosphere. I think that's quite familiar, right, to you? And then, of course, if you then look at the taxonomic diversity of the microbes, I think everybody is familiar with the symbiotic microbes, so the nitrogen-fixing rhizobia and the mycorrhizal fungi. But then if you look at the picture here, and I made a drawing comparable to the human microbiome, so you have the human genome, and then you have all the microbes with the genes there. So again, for plants, you have the archaea, we have the protozoa, viruses, algae, bacteria, and there's many nematodes here, fungi, oomycetes. And if you look at all the studies that have been published on the plant microbiome, there's still a big focus on bacteria, also fungi, but when you go to the other organisms, you see a sort of like a dilution. So there's a lot to explore there. So it's super exciting to see, what do we know and what is still to learn. So the who, where, what. So if we move above ground to the phytosphere, there's a beautiful initial review paper. There are more papers on this by Julia Forhold, in 2012, where she did some cartoons here on the complexity of microbes living on a leaf surface. You can see here. So they produce extracellular polysaccharides. They are involved in nutrient uptake and metabolic adaptation. So they produce surfactants, antibiotics. They also even produce plant phytohormones, pigments, usually to protect themselves for UV radiation and adverse conditions. And you can also visualize them. So where are they located? Either through electron microscopy, or very simple, if you have a leaf surface and you print it onto an agar, you can actually simply look at the distribution of these microbes on a leaf surface. Okay, so in terms of what microbes can do for plants, it's a bit of a simplification, but I like to sort of like categorize, humans like to categorize things into a box, right? And if it doesn't fit in a box, we usually have an issue. So here we did, that's also in an older review that I wrote together with Rodrigo Mendes. So we have the good, the bad, and the ugly. I've seen from the projects here, there's a number of people focusing also on, plant pathogenic fungi or nematodes. Also on the good ones. And then, of course, there's the ugly ones. Sometimes food products or plant products are also colonized by microbes that can actually cause disease. And there's a number of examples in literature. This was a picture from an E. coli outbreak of E. coli colonizing these little seedlings that you can buy in supermarkets for food. Okay, so I talked about the human microbiome, I talked about plant microbiome, but if you look at the similarities between those, and again there's a few papers on that. This was a paper that we published in 2015. The gut and the rhizosphere, for example, they are characterized by gradients of oxygen, pH, water, and there's also many different niches, right? So the rhizosphere is also called the gut inside out. They are open systems with a large area, and then they are shaped according to, very similar things like genotype and age. And also if you look at the two major phenotypes, so health and development and growth, also when you then plot, the mechanisms that are known so far for microbiomes, there's a surprising level of similarity here. Impact on physiology. Microbes can enhance nutrient acquisition, production of specific vitamins and growth hormones. And when it relates to health, microbes can protect against pathogens, they produce enzymes and antibiotics, and they also modulate the immune system. This is a bit of a simple cartoon. There was a recent review from Corné Pieters's group in Utrecht. I think it's still singing, huh? Yeah? You mean the speaker of the... No. I disconnected the speakers already.
So that the sound goes there and then...
Okay. This should do it. Okay, good. This is a more... No, it's still there. This is a more, mechanistic cartoon of both systems. So on the left here, you have the gut-brain axis. And you remember the table I just showed you with all the mechanisms there? That's listed here. So how specific short-chain fatty acids and specific hormones and neurotransmitters have an impact, on a distance from going from the gut microbiota to the brain. But also for plants, similar, mechanisms are emerging here. So you have compounds that are involved in, the communication between symbiotic microbes, rhizobia, and the plant. But also there's a lot of chemistry here, which is being elucidated. So the production of strigolactones, flavonoids, and then again there is some first evidence where you have transport of specific proteins to the shoot, which have an impact on shoot development, and also the fate of stem cells in the apical meristem. Yeah? So, so again here, lots of similarities. The microbes in both systems are usually quite different, but at the mechanistic level, there's a lot of similarity. Okay, so how to study microbes? This is what we have done yesterday in the practical, right? We have done plating, and like Victor already indicated in his lectures, there's a number of microbes that we cannot culture, or at least we are not able to culture them. And then there is the DNA, RNA metabolomics type approach. So which microbes are there? This is what we have done yesterday. We have isolated DNA from the same sample that we used for plating. And with that information, so if we would send that out for sequencing, we could have a look at the taxonomic diversity that is present in these samples using either 16S or ITS or 18S, depending on the organisms that you are interested in. And the computer practical today, we will go into how to do 16S amplicon analysis, right? You can also do RNA, and then you get a much better view of which microbes are active in the plant microbiome and which genes are expressed in that. You can also go to the protein level. That's called metaproteomics. And you can also directly go to the metabolites that are produced by microbes in the plant microbiome, and that of course is called metabolomics. And metagenomics, that is, you use the same DNA as we have isolated here, but then you do a sequencing, not of a specific amplicon, but you do shotgun sequencing where you sequence all the genetic information in your sample. And that will give you an idea of what kind of gene clusters are there, what kind of functional potential is there, right? So this is basically the toolbox, a culture-independent toolbox. Then something I would like to discuss, and it's mixed many times in studies, so you can look at the microbiome as a phenotype. For example, you can say, I'm interested to see if I grow different cultivars from the same plant species, how does that impact on my microbiome composition? So you basically look at the microbiome as a phenotype of your plant. Yeah? Or if a plant is infected with a pathogen, a nematode or a fungus, how does that impact on the microbiome composition? So you again look at the microbiome as a phenotype of that particular treatment. The reverse is, what kind of phenotype is associated with the microbiome? It's a different question. So that actually refers to how does the microbiome impact on the phenotype of the host? And if you go to, and you will see it also in literature, the literature references that we provided you, many times these things are intermixed. I will give an example of both systems. So a very nice recent review by Trivedi in Nature Reviews Microbiology talks about the microbiome as a phenotype. And in that review, they basically mapped the diversity of the microbiome at different plant parts. So the phyllosphere, the endosphere, on the leaf and on the roots, and then you go to rhizosphere and bulk soil. And again, there's here with fungi, and here you have the bacteria. And then if you compartmentalize that and you do a diversity measure, and this is the diversity measure, that's the Shannon index, which is one of the ways to actually quantify diversity within, and Victor will also give in his lectures later today other measures of diversity, because Shannon is not the only one. But this is just to highlight. So you see in the bulk soil, there's a high diversity. And when you move to the plant surface here, the rhizosphere, you can see that the plant selects, a specific subset of the microbes from that bulk soil. And if you go then deeper into the plant tissue, into the endosphere, you can see there's another level of selection there, because some microbes cannot enter the endosphere. And for the leaf, there's a bit of the same for the bacteria. So here you have the phyllosphere and then the leaf endosphere. This is for bacteria, and this is for the fungi. Similar principles apply there. And then, you know, again, if you look at, you will see it in the papers that we provided you. When you look at the microbiomes as a phenotype, you will get all these types of plots in papers. Usually here, a relative abundance plots with, this is at genus level, where, you know, the bacterial community is mapped according to the treatment. The pie charts, you know, so Luisa yesterday in the computer practical showed you how to make these. And then also here, a more, you know, principal component analysis where you try to see are there specific patterns in the treatments and how that impacts on microbiome composition. Now there's one thing you know, many papers, they look at relative abundances at the phylum level, right? And I just wanted to bring back some very, you know, so if you look at the taxonomic delineation of microbes or organisms in general, you go from phylum level to class, order, family, etc., all the way down to strain level, right? That's where we are. If you do that, so I put here a plant, a flower, humans, and an elephant. So if we make a classification based on phylum level, yeah, we can discriminate between plants and the animals here. But if we want to make a comparison at phylum level between a human and an elephant, yeah, we have to go down to order level to actually discriminate them. So think about that when you see bar plots in papers, you know, usually at phylum level. It does give a picture of, you know, does a specific treatment, does it sort of like give a change at phylum level? But at the functional level, it is, yeah, the meaning is very limited. And even at, you know, the species or genus level, this is a paper I was involved with many years ago where we looked at the diversity of a bacterial genus, Pseudomonas, associated with plants. And what we did is we sequenced the genomes of a large number of Pseudomonas species and strains within a given species. And of course, there is a core genome here, and then there is an accessory genome. So every strain or species has a set of genes which is, you know, similar to the other ones, but it also has a specific subset which is different. So at the functional level, it can mean that, you know, working at species or genus level will not even give you sort of like the resolution that you are looking for. So what we did with these, all these different Pseudomonas species and strains, we also mapped all the metabolites that they produce. So a set of antibiotics, surfactants, lipopeptides, iron scavenging compounds like siderophores. There's a lot of gene clusters for which at that time we did not know what they were, so we called them the orphan gene clusters. And then bacteriocins, so those are compounds that the bacteria produce, but they only have an impact on taxonomically closely related organisms. And again, you know, if you go again to the strain level, you can see at the strain level how they separate out. So for you, for example, if you are interested in microbes, you know, for a particular purpose, yeah, I hate to say it, but you have to go to the strain level. At the species or genus level, unless you focus on traits that are very common among a specific genus or species. But many times, you know, the detail is in, you know, the strain level comparison. Okay, so that was microbiomes as a phenotype. So how does the plant, how does a treatment impact on the microbiome composition? So let's go to how microbes impact on the phenotype of the host. So we have, of course, plant growth, development, and stress tolerance. I think that's quite well known. And then we have the plant quality trait. I will come back to that in a minute. And this is, of course, in terms of application, used in microbiome-associated plant breeding, nature restoration, healthy foods here, but also natural product discovery, and microbial biotechnology. So there's a lot of information that we get from microbes associated with plants that also have, a much broader application than only to plants. So how to study MAPS, so microbiome-associated phenotypes. I think during the inauguration yesterday, it was already mentioned that, the classic GE model, so the phenotype is your genotype times environment, right? So where the field is moving, and we published a paper on that a year ago, 2 years ago, where we added M. Now, in the classic model, M stands for management, but we used M here as the microbiome component. So actually, we made a model where the phenotype, Y, depends on the genotype, the environment, and the microbiome, and all the interactions between those. There is, Louisa yesterday mentioned about GitHub, where you can find scripts to calculate this. So you can build your own gem. If you're interested in the system that you're working with, what is the contribution of my microbiome to the phenotype that you have, you can go to GitHub, get the script, and simply calculate. So just to give you an example, if you're, and that's a super simple experiment, so you grow your plant in a natural soil with a particular stress or not even a stress, a nutrient stress or an abiotic stress, and you do exactly the same experiment where you grow your plant in the soil which is fully sterilized. Yeah? So there you eliminate your microbiome. So as a readout, you can then look at what are the phenotypes. So you can look at biomass, you can look at development, and that you can use here. So, so the host phenotype, and then you can put all these variables in here, and it will give you the relative percentage that the microbiome, based on this comparison, has contributed to that phenotype. Okay, again, coming back to, so to what extent do microbiomes actually influence this phenotype or the ecosystem functions? And then if you have a phenotype, which microbiome members and traits on the microbe and the plant side are key. And then when you go to the methods that we have available, it's not so super yet. So again, if you want to eliminate microbes, you can go super crude. You can sterilize your system, right? But that means that you basically eliminate all the microbes. It does not help you to pinpoint, the members in the microbiome that contribute. And to do that, some groups, they use specific biocides that eliminate specific members in there. It works, but again, the level of fine-tuning, again, think about going to the strain level. That's super difficult here. And then we have the microbiome transplantation. So you take a microbiome from a system and you transplant it to another system, and then you look for the phenotype. And one of the things that we have done in the context of disease suppressive soils is that we have, you can transplant the microbiome to a system and then the plants are protected. And what we have done, and I'll show an example later, once you have that phenotype, you can, with the transplant, you can start diluting your transplant at small steps. So you dilute it twofold, you transplant, you look for the phenotype. You dilute it fourfold, eightfold, etc., and you look for the phenotype. And then basically what you do, and that's what it's called tipping point, you look at the point where your phenotype is lost due to the transplantation. So you basically simplify your microbiome community by dilutions, and then around that tipping point where you have the phenotype or you don't, you can do the sequencing and then see, which microbes or which traits did you lose, yeah, through the. And then once again, phenotype for me is always the first and foremost. Is there a phenotype associated with your microbiome? If you have a phenotype, you can do the sterilization, etc. And then once you have that, you can do the profiling. So by, like we did yesterday, you isolate the DNA, you do the amplicon, metagenome. There's also network inference. And then you go, from that data, you go to the culture collection that you have established from that same sample. And then you try to pick out the microbes. You reintroduce those microbes either alone or in a synthetic community to recreate the phenotype. And then once you have identified the microbes that do that, you can go into more mechanistic work. You can look which genes are important, which metabolites are important for that phenotype. Yeah? Usually that's a lot of work to go all the way to that level. So I've talked about, studying maps by perturbation, so heat treatments, so temperature treatments. You can do selective temperature treatments. You can go to 55, 65, 80 degrees, and even higher. Dilution, I just talked about that, and specific biocides. So you can, for example, add a fungicide to your system, to the soil, to eliminate the fungal, contribution. You can add a specific antibiotic to eliminate a specific group of bacteria in your system. And again, then have a readout in terms of phenotype, what that treatment means. So this is an example of the dilution or the intensity of perturbation. And the curves that you see here, so here you have the microbiome-associated phenotype. So if your sample is not perturbed, here you have the maximum phenotype. And when you start to add biocides, or you dilute, or you have a specific heat treatment, you can have multiple response curves like this, or a linear one, or this is, a typical response curve that you get. And like I said, at this point, there is sort of like a tipping point in the phenotype, and around that tipping point, you can do the microbiome analysis to pinpoint the groups that are of interest. One example, this is an example from a Korean group. So what they did, and I'm sure that for those of you here who are very familiar, if you go out in the field, sometimes in a field you see these patches of plants which are diseased and patches that are healthy. And then it's like, okay, so they observed exactly the same for tomato and for a bacterial disease called Ralstonia, caused by Ralstonia. And they also observed plants in a field site where some were heavily infected and others, which was very close, they were super healthy. So what they did, they collected the rhizosphere environment from these tomato plants, and they first did a profiling. So, what are the differences in the microbiome between these two phenotypes, so the diseased and the healthy ones? And they observed that in the healthy ones, there was a higher relative abundance of actinobacteria and bacilli, firmicutes. Okay, that's a nice first observation. It can, point you in a specific direction. And then what they did is they took an antibiotic called vancomycin, and vancomycin works against gram-positive bacteria, against actinomycetes and bacilli. So they took what they call HRS, which is the healthy rhizosphere sample, and then the DRS, that's the diseased rhizosphere sample. And they started treating those with vancomycin. And, and then once they did that, so they did the treatment for 30 degrees, 3 hours. They washed out the vancomycin after the treatment, and then they basically transplanted these rhizosphere fractions to a pod system, a test system, where you basically test the effect of those, rhizosphere samples on disease. And they introduced the pathogen. So that's all normalized at that point. Have a look at this paper, there's much more in there, but what's super interesting is that indeed they could confirm that when you treat a healthy rhizosphere sample with vancomycin and you eliminate or at least reduce the density of these gram-positive microbes, that disease is back again, right? Quite straightforward in terms of treatment. It's usually, it helps you to go like, okay, which part of the microbiome should I look at for my PhD and master work? Then transplantation, mentioned before. I think everybody is familiar with this poop transplantations. Maybe not. Okay, so there's a number of people who suffer from chronic bowel diseases and Crohn's diseases, and what they have started to do is they have started to transplant poop from healthy individuals to these patients. Yeah. It seems very, this is, if you go back to the literature already in the old Chinese medicine, that was a treatment that they actually used to do that. So, but what they basically do is they transplant, a healthy microbiome to a diseased microbiome. And usually what they do is the people that suffer from this Crohn's disease, they give an antibiotic first, so basically to reduce or to eliminate most of the microbiome that is there, and then they transplant the healthy ones to make the establishment of the healthy, easier, right? Because otherwise you introduce a microbiome and it has to establish itself in competition with the microbes that are already there, okay? Yeah. And just to make it, this is in Dutch here, but there's even a group, you can look it up on the internet, it's called Dr. Armpit. What they do is they look at the microbiomes in your armpit and how that impacts on how you smell, right? And they have also they have started to transplant microbiomes from armpit of people who smell nice, but I mean, that's quite subjective, right? Who smells nice, but to persons. So don't be surprised, and in fact, I had a look at that. There's going to be deodorants, etc., where there is sort of like a probiotic, a microbe added, just, to promote the smell. Now, I mean, that's, funny and all nice, but what is super interesting here is if you look at these transplant studies, going back to the very original slides that I said, genotype is very crucial for the success rate. So they did studies on this with the transplants with people that are genetically very related and people that are genetically unrelated, and the success rate of these types of transplants is higher when you have people that are genetically more related. So they did twin studies with the armpit here, where one of the twins smelled quite bad and the other one smelled good, and they could actually recover that for quite a while. Okay, good. In terms of plants, we also did studies with soils that are suppressive to a specific soil-borne disease, in this case rhizoctonia, on sugar beet seedlings. So this is plants growing with the pathogen in suppressive soils, and these are plants growing in a soil which is physically, chemically very similar to this one, and you can see that all these plants, they die, right? The transplant is super simple, so if I take a little bit of soil, 10% of this soil, I mix it into this one, and you do the assay again, you go from this phenotype to this phenotype, or close. If you sterilize this one at 50 degrees or at 80 degrees, you go from this phenotype to this phenotype. Yeah? So, and having this sort of like transplant, so if you know that a transplant can confer a phenotype, again, you can, like I said, you can take the transplant and start diluting it, and when is the point that it does not give the phenotype anymore? And then again, sequence around that point. In the institute where I work, there's also a group, super interesting. So in the Netherlands, we are trying to give agricultural land back to nature, right? So to avoid, the pollution with fertilizers, etc. And the group of Wim van der Putte and Martijn Bezemer, what they did is they did transplants from natural ecosystems into these agricultural soils, and the speed by which these agricultural soils are restored back to natural conditions is faster when you do a transplant. So that's, yeah, and you can imagine the scale here. I mean, that's transplanting huge, so they transplant the soil in the top layer. Then a last example, and then I think we can, I'm also looking at Victor, I think we can go for cookies and coffee right after this one, and chocolate. There are some really cool new developments in how to study microbiome-associated phenotypes. And that's by phages and by CRISPR-Cas. Phages, I think you are familiar with that. You can use phage therapy, so lytic phages, to control bacterial diseases, right? In the microbiome field, what they are trying to do now is also to get lytic phages of specific groups within the microbiome that you can eliminate, and then basically look at what is the impact of elimination of a subset, again, on the phenotype. That's one thing. And then the other, which is in full development now, is to use CRISPR-Cas, so to have the guide RNA and the DNA nuclease, the Cas9, as a way to eliminate specific members of the microbiome, or even target specific genes within the microbiome. Yeah, and I've given a few very recent references here, which this is a review, super nice review by Ramachandran here and Picard, where how to use CRISPR technology to edit the microbiome. This is also a really nice paper on the gut microbiome. And if you're not fully familiar with the CRISPR-Cas, I also provided a link here where you can look at this nice movie where CRISPR-Cas is nicely explained and how you can use it for editing genomes and also microbiomes. So with this, like I did with the human microbiome, so what is known and what is unknown, I can tell you there's so much unknown yet. So we know actually, the tip of the iceberg. We know quite a lot about bacteria, a bit less about fungi, but then if you move to viruses, which is super upcoming, if you go to archaea, protozoa, fungi, oomycetes, and nematodes, yes, there's quite a lot known more from a pathogenicity perspective, but how they contribute to, for example, beneficial effects, also there's a lot to learn. So there's super nice opportunity for you to discover new things here. So okay, with that, I'm going to stop here. Are there any questions? Yeah. Sorry, I don't get the question. The tipping point, right. So with the tipping point you can do two things. So with the transplant, right, you transplant, for example, the suppressive soil example, right? You transplant 10% of soil, you get the phenotype. If you take 5%, do you still get the phenotype? So yes. If you take 2.5%, do you still get the phenotype? If you go to 1%, 0.5%, 0.1%. So that's how you do the assay. So you do that in one shot, and then in the greenhouse you see, okay, here the phenotype is still there, so undiluted, 10 times, etc. And at the point where you lose the phenotype in terms of protection of the plant, around those 2 points, it's because that is, so if the tipping point is between 1 and 0.5 or 0.1%, you basically sequence around that tipping point, because that's where the phenotype goes from good to bad. You can do the sequencing, but you can also do what we did yesterday in the lab. You can also plate on a number of media and then basically see, okay, what are the microbes there. Because dilution to extinction is not a simple phenomenon, because you can imagine if you dilute, your diversity becomes less, but the relative abundance of specific microbes, because you basically change the composition, also starts to change. So it's not a super trivial, like, a linear type relationship. But that's the way to, actually it's the way to minimize the complexity that you have in the full system. Yeah. Does that answer your question? Okay.
The formula genotype. The genotype formula, expanded formula.
Jump.
Jump.
The jump, yeah, the GEM, yeah.
So what is the meaning of the last letter small letter b?
Yeah, that's that's a good question. That's what they call a stochastic variable. Basically, that's sort of like a variation that cannot be explained by the G, the E, the M, or the interaction between the two. There's always an additional.
So that
Exactly.
has to be error.
Sorry. It does not stand for error is no, it's not an error, but it's a stochastic variation that always occurs in the system. Yeah. Yeah, we can also interact at the coffee, right? Or if later today a question pops up, just ask me. Okay. I'm just looking at the schedule, Kumar. Shall we have a break now, right, Victor? Yeah. So we have a coffee. We brought some chocolate and.
We have chocolate. Coffee is like you control government like chocolates you can have. No problem.
Sure. Yeah. Good. And then, okay. So we have the coffee at 11 then. Victor, would you like to? What do you prefer? You want to have a break now and then have one run, because the coffee and tea will be there at 11 o'clock. Yeah. Yeah. Then maybe 45 minutes is enough, Vic, for your first part of the lecture. Yeah, but logistically, the break is organized at 11 o'clock.
Maybe tomorrow we can see that. Today.
Yeah? Okay. Then we'll do it like that. Yeah, so that was a teaser with the chocolate, and now we have to wait, right? It's, sorry. So again, the slides of the lecture will be made available, right, on the shared drive.
let's get started, right? So, well, after the nice lecture from Josh, we are gonna start going deeper and deeper on the 16S amplicon analysis and how do we get to the 16S amplicon and so on. Okay, so we call this lecture Microbiome Technologies and Analysis 1. So there will be a 2 and a 3, okay, where we will be looking at different methodologies like metagenomics or genomics. Okay, so just to give you an idea where we are, I think you know where we are already, but yesterday we did this part, we look at the diversity of microbes, we cultured them, we isolated the DNA, I don't know, the quality was not so good, but we did our best, then we moved to the NGS sequencing now. So yesterday, Kumar, when we finished the isolation, he took the DNA and then he ran to the sequencing company, and then now he's sequencing all our samples, our 10 samples, right? Kumar?
Yes, we did, yeah.
Perfect. Okay, so then today we are going to be looking at, well, when we receive the 16S amplicon, so the raw reads, what do we have to do? And we will start with the taxonomy. Also, we will look also at the differential abundance, but mainly focusing on taxonomy. Are you with me?
Yes.
Yeah? Perfect. These are the learning goals of today. So we will start with the toolbox. Actually, it's the same picture than Josh already showed. So this is the different tools that we have for metagenomics, 16S, metatranscriptome, and so on. There will be a part where we will be looking and, okay, so, well, now we know the tools that we have. So we have all the information. So how do we decide, what are we going to do, or which method to choose? We will look a bit more in detail into 16S, ITS, and 18S. I think you are all familiar with these names. Then we will be looking at clustering versus OTUs, so versus ASBs, so OTUs versus ASBs, and we will go more in detail in a bit. Then we will move into, well, we get the OTU table or the ASB table, Luisa yesterday showed how we do tables in R, right? So when we have an OTU table or an ASB table, basically it's the same. We get a lot of counts, and then the treatments, and then the OTUs, okay? So we will look in this part at, well, how do we analyze this data, okay? And in the afternoon, Guillermo and, well, Luisa and me, we will be doing hands-on, how to do the analysis and so on, okay? Right? Well, let's get started. This is the second time that you see this slide today because Josh already showed. So, well, when we are looking at microbial communities, we have, well, 3 main questions. So, which microbes are in there? How are the microbes doing the functions? And what is the genomic and genetic potential, okay? So, to answer these questions, we can use different tools, And the different tools are listed here, okay? So, we have to answer which microbes are in there, we can do amplicon sequencing. So for that, we take a fragment of the DNA, of the genomic DNA that is conserved among different bacteria, archaea, and so on, and then we sequence, and then then, well, we analyze. Okay? To see what is the potential of those microbes, we do metagenomics. Okay? When I say metagenomics, I mean shotgun. So we take a sample of DNA in a particular environment, soil, nematode, insect, whatever you have or whatever you are working with, and then you sequence everything, all the genomes, not a fragment, yeah, that is conserved among all the bacteria. You sequence everything that is in there, okay? And then to know how or what the microbes are doing in that natural environment, in that particular environment that you are working in, well, we have many other different omics. For example, RNA, which will be metatranscriptomics, we look at the functions. Then proteins, we look at metaproteomics, and then metabolomics, yeah? And then we do LC-MS/MS, and then we look at the whole metabolome of that system. Are you with me? Yeah? Okay, but now we know all the sequencing technologies or the tools, yeah, to study microbiomes. Okay? So before selecting a tool, yeah, we need to know how to choose it, right? So, for example, I don't know in what you are working, but yeah, if you are working with soil, you have high diversity, right? So then you will need to go more in-depth sequencing. If you are working with endophytic bacteria, so bacteria living inside a plant, the community composition will be less diverse, right? So then you need to, maybe you don't need to sequence that much, okay? But let's go question by question, okay? So first of all, what is the research questions or research question or hypothesis that your study is aiming to address? Yeah, so for example, if you want to look at the taxonomy, yeah, you only need to look at what is the microbial composition, then you do simply a 16S amplicon or 18S or ITS. Why do you need to do a metagenome? Metagenome will be extremely expensive, yeah, and then, well, you only need to do 16S amplicon that it's cheaper, okay? So that's the first thing that you need to think. So then yesterday we did DNA extraction, and well, what is the biological sample that will be analyzed? There are many different samples. It could be fecal samples, soil samples, water samples, etc. So when you are doing the DNA extraction, there could be possible contaminants. And yesterday, yeah, when we did the extraction, well, some of the samples were really low concentrated, and we saw that some of the ratios were very low. Was it like that? What could be the reason? Well, I don't know exactly, but could be that that soil has something that could be interfering with the measurement of the DNA concentration, okay? There could be many reasons. I'm just trying to find an answer right now. Okay, so as I said at the beginning, what is the expected complexity of the microbial community that we are working with? So if we are working with soil, yeah, we will have a high diversity, yeah, a lot of different microbes. If we are working in the rhizosphere, we will have a lot of microbes, yeah, a lot of different microbes, but less than in the soil. Then if you move to the endosphere, phytosphere, and so on, the diversity, yeah, will be reduced. Are you with me? Perfect. So then also, I think you are all familiar with what is Illumina, what is nanopore, what is PacBio, and so on. So what are the limitations of the techniques, yeah, that we are using? For example, for 16S amplicon metagenome, we usually, yeah, use Illumina. For genome sequencing, we use nanopore, and all of those techniques, they have different error rates, and they are designed for different purposes. Okay, so you need to know exactly yeah, which technique to choose, and then, well, at the end of the day, you will be spending less money, and well, you will be, yeah, more accurate. Extremely important, controls. Yeah, so you need to have the appropriate controls because, for example, imagine that you are interested in, well, you have your plant, and then your plant is infected by a pathogen, but then your plant is resistant. But, well, yeah, how do you pinpoint specific bacteria involved in that resistance if you don't have the control? The control is the non-inoculated one, right? So you really need to think in your controls. Okay, and the last question. In the afternoon as well, Guillermo will show you one of the methods. So how the data is going to be analyzed and interpreted. So I think that's something that you also need to be aware of. So, for example, there are many methods, metagenome-seq, DSEQ, HR, you name it, and they are all valid. Yeah, you can use them. There is no problem. There is nothing wrong with any of them, but you have to think what are the possible biases, yeah, depending on the method that you use. Okay? Still with me? Okay, but there is a bit more. Extremely important, very, very important. What about the metadata? Do you know what is the metadata? You all know? Yes? No? Yes? Raise your hand. You know? No. Okay, good. So the metadata, yeah, is the information about your samples, okay? Where was the sample taken? What type of soil? How many replicates? Well, all that you have here, yeah, you could add it to your metadata because as much information as you have, then, well, it will be better for later analysis to explain the difference that you see between your treatments, okay? So then, yeah, date and location where you take the sample, that's very important. Yeah, experimental metadata, controls and replicates, physical and chemical properties of your soil or whatever environment you are working with, etc. There are so many things that you can add to the metadata. All the details, if some samples fail or if you saw something different when you were isolating the DNA in one of your samples, all of those things is good to make notes, yeah, in your metadata, and it will be very helpful for later analysis. I really want to emphasize that, yeah, data, like it's meaningless. So if you don't know where they are coming from, if you give me, yeah, FASTQ files from your sequencing microbiome, the microbiome that you sequence from your plant, well, if I don't know any other information, well, I can get the data, but what do I do with it, right? Yeah, so then, of course, microbial communities, yeah, as I say here, are highly adapted to their environments. So to make sense of the results, you really need to know, yeah, what is the metadata. Okay. Good. There are well, and also when you are going to publish your data, so there are several repositories. You have the KITA, the EDI, or the MIXT. So all of these repositories are going to ask you in advance before you upload the data, the FASTQ files. So all these repositories are going to ask you for a detailed metadata. So if you start in advance, it's better if you know this before starting. Okay. We are still together? Yes. Then we have seen all the tools that we have, right? So, and when we get our DNA, we send for sequencing, and then there are two main techniques let's call it like that. So we have amplicon sequencing and shotgun metagenome. I'm going to repeat this a lot of time because I really want you to differentiate between them. So amplicon sequencing is going to be using a fragment that is common between bacteria, archaea, eukaryotes, and so on. So it can be 16S, 18S, or ITS, or any of the housekeeping genes that is shared among them. Okay? And what kind of information do we get from 16S? We get just taxonomic information, so which bacteria are in there. Shotgun. Shotgun, we get everything. Okay? So we extract the DNA, we break down, we get the fragments, and then we sequence everything. We get the taxonomy of every read and the functions. Okay? So today we are going to be focusing mainly in the morning and in the afternoon with Guillermo. We will be focusing in 16S amplicon. Again, the three main questions. Yeah. So what we are going to see is who is in there using 16S, 18S, and ITS. Yeah. And we will mainly talk about 16S because, yeah, my background is more in bacteria, and I don't have much experience with 18S and ITS, but I will also tell you the primers that are used for ITS and so on. So, first of all, you know that it's 16S already, but yeah, how to select for a gene marker? How to know that a gene marker is good for amplicon sequencing? Well, the first thing that you need to check is the genes that you select have to be, so it has to be everywhere and it has to be important for the list of all microbes. Then these genes have to contain two features. The first feature is it needs to have a conserved region. And the second one, it needs to have a variable region. Okay? Conserved and variable. So the conserved region is common among all microbes. Okay? Bacteria, archaea, and so on. So then in that way, we can generate primers in those conserved regions and then amplify that fragment. Okay? Then the variable region is different among all taxa. And that's what we are going to evaluate when we do the sequencing, because they will be specific for each microbe to a certain extent. Is it clear? Okay, so I already told you, but we know that the marker of bacteria is the 16S gene. Okay? So the 16S gene, the 16S RNA gene, it's present in all species. It's an ancient gene, and yeah, it contains variable and conserved regions alternated. And actually, it is an evolutionary chronometer. There are other genes as well, like housekeeping genes that you could use, but they are going to be a bit more specific and difficult to generate primers. Yeah, like for example, I put here RPO-A or GLB. Yeah? So you could also use those ones, but the most common one is the 16S. Okay, so this is the 16S region. So here you can see in gray the variable regions. You have from P1 to P9, and also the conserved regions in green. Okay, so when you want to design the primers for the 16S amplicon, the logic would be to design the primers in the conserved regions. Okay? So here I put you this paper. Sorry, it's a bit blurry. But yeah, so in this paper from 2013 from Pfizer, so here they look at the efficiency capturing the microbial diversity in using different regions, different primers. So, well, you can see that these two regions, I cannot read which one it is, but I guess it will be B1, B2, or B3, B4. Yeah, you get more or less the same microbial community composition, and when you use others, yeah, you get different microbial composition. So here in the y-axis you have the relative abundance, and here the different primers, and well, here what you see are the different taxonomy, the taxonomy composition. I think, well, you already saw several plots like this videos. Is it clear for all of you? Yeah? Perfect. So the most commonly used and the most recommended region to use is the B3, B4. Okay? So this is the most common region for amplicon sequencing. But you can use other regions depending on your purpose. So that's why the questions at the beginning were very important, because you really need to know what you are looking for, yeah, in advance. For example, is there anyone here working with endophytes, endophytic microbiome? Nice, perfect. Well, there are some primers that are more specific for endophytic microbiome. And those ones, well, those are from the B5 to the B7. Okay? So you could use those primers to avoid cross-contamination with chloroplast and mitochondria. Okay? Yeah, so maybe later, if you need a bit more information, I can give you some reference for that. Okay? Is clear for everybody? Okay, next marker. Wow, this is really bad. I almost cannot see. Okay, next marker is the 18S. So this is part of the small subunit of the eukaryotic ribosomes. And well, you know, it's a basic component of the eukaryotic cells. It's structural for the small component of the eukaryotic cytoplasmatic ribosomes. And well, it's very important for maintaining the structure of this of the ribosome. So yeah, this is falling in the category of, well, it's very essential for the living microbe. Okay, and it's the same than the 16S. You have different regions, and then, well, here in black are the variable regions, and then in white you have the conserved regions. And well, when you contact a company, they will tell you, okay, what do you want to do? You want to do 18S, we have available these primers, and then you have to choose. But of course, you need to know which region you want to amplify. Then, well, I forgot to say that 18S it's well, to look at all, yeah, eukaryotic organisms. So here you can look at protozoa, you can look at fungi, oomycetes, and everything. So when you want to go a bit more in detail and look a bit more into yeah, the diversity of a specific taxa or group, in eukaryotes you use the IPS. The IPS is the spacer located between the small and the large rRNA subunit genes. And it's, as I said, it's most often used in fungi, but you can use also for other eukaryotes using other specific primers. Okay? Okay. Clear? Yes? Okay, so let's, I did this slide yesterday, thinking that it would be nice to give you also an idea of what is good and what is not so good. Yeah? So what I call it pros and cons. So again, I'm not gonna stop repeating myself. So 16S only provides a snapshot of the taxonomic diversity in that environment, yeah, in that you are working with at that particular moment. I put here is inexpensive, but yeah, well, I mean, it's more affordable, it's cheaper than metagenome sequencing, and well, also something that, yeah, I didn't say, but working with metagenomics, it's computationally very expensive. So you need powerful servers to work with metagenomics. So working with 16S, thinking the way that you are working with a small fragment, yeah, 500, 300, 400 base pairs. So then it's easier, yeah, it's less expensive, it's less computationally expensive to work with 16S amplicon. Although Guillermo will tell you today, so we have pre-computed most of the analysis because to analyze 16S, you need you need a server depending on the method that you are going to use for the analysis. And we are using data too. I am already telling you, but yeah, I will go more in detail later. Well, something that yesterday I talked to some of you, so we got really low amount of DNA, and well, I was not worried about it because if you are doing metagenomics, you need high amounts of DNA, but when you are doing 16S, with really little amount of DNA, you will be able to get what you are looking for. That is the taxonomic composition. So it's, I think it's in the order of picograms of DNA. Although the company always asks you for, around 10 nanograms per microliter, I think. Are we okay with time? Yeah. Are we okay with the time?
Yeah.
Okay. Cons. Well, just already mentioned this morning, but yeah, it's not that good for strain level identification. And well, I think you can understand that, yeah, the 16S RNA fragment is around 1500 base pairs, and we are sequencing only around 500. And when we do the trimming and the quality filtering, we end up with 300, 350 base pairs. So we are trying to identify microorganisms, yeah, the taxonomy of that microbe, that specific microbe, using only 350 base pairs. So you can imagine that going deep into species level, it's going to be a bit difficult. Yeah, so we have to, stay a bit like maybe at genus level or even family level, or well, as Josh was showing today, sometimes people look at even phylum level if you have big difference between your treatments. Okay. Well, this relates to the questions that we already had at the beginning about, well, how to think or how, to choose the right tool. Yeah, so there are many different bias depending on the primers that you choose. Sometimes you will get more archaea depending on the primers that you choose. Sometimes you will get more protobacteria and so on. Well, many other technical artifacts that I am not going to go in detail, but you always have to consider that. Okay. So we have seen already these three first points. Yeah. So do you have any question? Is it clear for all of you? Yes?
Shall we give a break up to this point?
The complexity, so I think we started low complexity. We are going to have peaks of high complexity. So it's up to you guys if you want to have a small break. Or
I could see, kind of, reflection.
also, the amount of information is a lot, so I think
We can start the clustering and the ASP is maybe up to the break. Is that fine?
Yeah, no, I'm fine with anything. Yeah.
Any questions? Yeah. Also, the tea is already there, but you can see that confident. Yeah. Exactly. Please, there is a question there.
of microbiome, if it is better to use V5, V7 to eliminate all those chloroplast and mitochondrial genomes, why V3, V4 is most commonly used, sir?
Well, because not all the people work with endophytes. So yeah, I love endophytes and I love to work with endophytes, but not everybody is working with endophytes. Most of the people is working with gut microbiome or soils or rhizosphere and so on. But also another reason why B3, B4 is more widely used is I think 100% sure.
Yeah, 515 to 8.
I think it's this one, B3, B4. So you are covering most of the diversity of prokaryotes. So here you get more anazyme and more, a better picture of archaea. So I think that's one of the reasons as well. But yeah, it depends also a lot on your research question. Yeah, what you are looking for and what you want to achieve for your research. Another thing is that that's a bit more complex, but when you do 16S amplicon, if you use any other region, that doesn't mean that your study cannot be comparable. Your study can be always comparable with others that use different regions, but then you use another approach, yeah? And this approach is basically you have a reference database where you have all the 16S amplicon, and we have done it in the past. I can give you some reference where you map your reads to the reference, and then you get the counts regardless the region and the taxonomy. Okay, I can give you some reference of those meta-analysis if you want. Yeah. I see another question there. I cannot listen. I'm gonna go down.
Why ITS is preferred over? Why ITS is preferred over 18 years?
In Punjab.
In Punjab, UK.
Well, that's not my field of expertise. I'm sorry for that. I just gave you a snapshot of what is out there. But my guess and what I have read is that the primers that are commonly used for ITS give a better view of the diversity of fungi. Yeah, so then if you are looking for another type of microbe, another type of eukaryote, I think you should look a bit more into detail to see if there are specific primers. Yeah, because I know, for example, that 18S will give you a better overview for all eukaryotes like protozoa, fungi, oomycetes, etc. Okay, but if you really want to use something more specific about IDS or 18S, you should look more in detail at the primers.
Sir,
Yeah.
sir, is there any specific reason that whole 16, V3, V4 is preferred than whole 16S sequencing? Sir?
Okay, the question is you have the full length sequencing now. Some companies are offering it anyhow. The full length 1500 base pair. Now the question she's asking whether is there any specific advantage of using V3, V4 over the full length?
Well, instead of the whole region.
Yeah.
Yeah,
the
The old
entire
science
region.
sequencing, right?
I mean, if you have the money, I would use the full region.
Exactly. Yeah. It's a cost consideration.
Yeah, exactly. But I think you have to consider, you have to go through all the questions and then see, what is your research question? What do you need to know?
Exactly.
Yeah, and then because if you want to go to strain or a species, then maybe you should consider indeed doing the full 16S. Myself, I never did full 16S. I don't know Jos group. Well, I think we had some data from full 16S, and of course you get a better resolution, but indeed my recommendation would be, okay, so think twice what you need, because I'm sure that full 16S is going to be much more expensive. Yeah.
it's moving.
Yeah. Also, we will see in, I think the last lecture we will be talking about metagenomics. When you do metagenomics, you can do also binning. I don't know if you are familiar with binning, but it's basically building draft genomes out of your metagenome. Yeah, and well, I'm gonna tell you already, it's not perfect because life in general is not perfect. But it gives you a better idea of what you have in there and what are the functions. Yeah.
Okay, we can continue because outside also is going to be here only. I think let's have a break, 15 minutes. Come back at 11:15. We thank yours and team for bringing some delicacy from Dutch. It's also served outside. Have a taste of some of the cookies and sweets from Netherlands. Now let's have a break.
from the Netherlands. Yeah, we're great. Okay. Well, I heard from Jost and Luis and Guillermo that some of you have problems installing some of the packages for the afternoon session. So I think I will be done, in an hour more or less. So then, before we go for lunch, we will do a round and then see how we can solve those problems, and if we cannot, we will find another solution, and so on. We will improvise. Okay? So yeah, now I am wearing this. I'm sorry for the people online. Before I was moving, and I hope that you could listen everything. Okay. Let's get started. So after the coffee, now we have sugar in our blood again. So we have seen already these 3 points: the toolbox, deciding how to do it, and we went into details of 16S, 18S, and ITS. Okay. So now we are gonna increase. Yeah. Thank you. Thank you. So now we are going to increase a bit the level of complexity. So please, if you have any question, raise your hand or your voice, and then, ask, please. So we are going to continue with the clustering or OTU and versus ASVs, okay? Then we will go to microbial diversity, what it's called also alpha diversity. We will see the difference later. Then beta diversity, and then microbiome data analysis, differential abundance, and some visualization tools. So everything that we are going to see here and what we already saw is in line with the practical this afternoon by Guillermo, but we all will be around. Okay, so when you do sequencing, this is what normally happens, okay? So it's a beautiful looking into the mirror, and he thinks that he's a lion, but yeah, this is the reality, okay? Then all of the issues that we have are try to bring us part of the mirror to see indeed what we are, and not the lion that it's in the other side. Okay, so this is more or less what is happening. This is the reality. Yeah, these are the true sequence. You have this sequence here, this one here, and this one here. But then when you do sequencing, due to the PCR cycles and many other issues, what you get is a kind of disturbed reality, a blur. Okay, so then you get this OTU as well. Sorry, this sequence, and this sequence. But then it's kind of diffuminated, so you get also many other sequences. Okay, so we need to correct that. Yeah, and over the past years there have been different methods to correct and to work on it, to work with that. So I'm listing here the different methods. Okay, so what we have to do indeed is clean our data, and manipulate the raw sequences. Okay, so in the past, I'm going to give you all of these slides. You don't need to take pictures, guys. Yeah. So so we have the what we have to do is two main methods, or moving noise, and this is the newest one. So when we are doing the clustering, we call OTU, Operational Taxonomic Unit, that you already saw it yesterday, but also today in the lecture of yours, and now I am explaining, and I am going to go a bit more in detail in what is an OTU, or Operational Taxonomic Unit. The first step when we are doing clustering, so getting OTUs, is remove the noisy sequence and reduce the amount of sequence to process, okay? So we are discarding data, basically, yeah? Data that it's, well, low quality, or it has mistakes, and so on. And this afternoon, Guillermo will go through this process. The most important is that, yeah, when you do OTU clustering, you are clustering into a given threshold, and you put that threshold. You decide what you do with the threshold. Yeah, normally we do it at 97% similarity. So the sequence that will cluster together, similarity. And well, it's up to you what you decide to do, but you have to really be sure. Yeah, well, based on the questions that I showed you at the beginning of, well, what you have to decide to proceed, okay? So there are different methods and algorithms to produce OTUs, yeah? And we are gonna look at all the methods, okay? So we have the closed method and the open reference method. And in the next slide, I will explain a bit more about it. But basically, one is that, well, you map the reads into a database, and the ones that are not aligning, you discard. And the other one, you map the reads to the database, and the ones that are not aligning, you create new OTUs, okay? Then the other technique is removing noise or correcting, yeah? So that's ASVs, amplicon sequence variant. So here, our purpose, yeah, and this is the newest method and what everybody lately is using. So what we are trying to do is to find the cleanest sequence, removing the errors or correcting the errors, okay? So we correct and discard a super noisy sequence. And some examples of algorithms that are doing this are Dibbler and Data2. Yeah, and Data2 is one of the most used, and today in the afternoon, Guillermo, well, will show you how to run Data2. I think I didn't mention here, so for clustering OTUs, there are many algorithms, many, you name it. So I just put here several of them that I used in the past, like uparse, uclust, or cdhit, yeah, but they are all based on this percentage of similarity, 97%, 98, 99, wherever you decide. Clear? Okay, so how do we generate the OTUs? Remember the cat and the lion? So here we have our samples. We have the green, the red, the blue, and the less blue, yeah, or gray. And then when you do sequencing, the amplification, this is what you get. You get, of course, yeah, the two ones, well, the four ones that you are interested in, but you get also a lot of errors and mistakes and spurious reads, okay? So we need to correct that somehow. And that's what the methods that I have been talking to you, that's what they do. So a way to correct them, to produce these OTU tables or ASV tables, is doing clustering. And you cluster them based on 97% similarity, yeah? And this is what I'm representing here, okay? So what you see here is that all the OTUs that were in this centromer, so they are clustering together, so they will belong to the same OTU. And the same here in the blue one. Is it clear? Are you all with me? Okay. Again, to generate OTUs, we have these two methods. Close reference, yeah, we have to cluster with database sequence, and then open reference, we need to use a database, yeah, and then de novo sequencing, or that not hitting with the database, we will create new new OTUs. Okay, so we are going to go one by one, and I'm going to give an example. I hope you can see. Yes? Okay. So we get our sequence. Yeah. Yesterday we did the DNA extraction. Kumar sent for sequencing. Yeah. We get our sequences. We have the reference database. Yeah. We map to the reference database, and then we have two possibilities. That our sequence doesn't have a hit. If our sequence don't have a hit, they are discarded. And then we focus that we generate our OTU table. Okay? Open reference. Again, we get our sequence to the reference database, and then out of the closed reference, we get them, we get the reference hit, and then the ones that fail to hit, we generate a de novo OTU, okay? A new OTU. And then we generate our data frame or OTU table. Clear? Okay. I don't want to mess up, but this is a comparison of all the methods for the clustering. This is, you cannot see, it's not good. Maybe I have to improve the quality of the picture. But so basically what it's doing, it's comparing all the methods of clustering that are available and then comparing it in studies. And well, I will refer you to the paper if you want to read a bit more about it. I think it's quite interesting. But so the conclusion is that most of the methods, they give you pretty similar results, but there are some difference among the methods, okay? Which one is good, which one is bad, I cannot tell, so you have to evaluate by yourself. Well, now we have got the OTU table, okay? So we have the OTUs, we have OTU1 and OTU-X. And then we have our different treatments. We will see how it looks like an OTU table. Now, next step is to get the taxonomy. We have our clean sequence. Now we need to map to the reference space and get the taxonomic composition. So this bacteria, this OTU, it's a bacteria and it belongs to Pseudomonas or Burkholderia or whatever, okay? So there are many different databases, and they have all pros and cons. We use a lot Silva database. I think it's a good choice for all of you. Green Genes is also good. I kind of like also RDP. You can take your 16S sequence and then map it into the RDP, and then you will get a score and a probability of this bacteria is mapping with this other bacteria from the database and so on. So I think they are all good, but normally we use Silva database. And this is a comparison between different taxonomic databases. So if you want to know more, so here is the paper. I will pass you the slides so you can go a bit more in detail if you are interested in that. Okay, so we have seen the clustering, but we didn't see yet the ASBs. Okay, so ASBs is standing for amplicon sequencing variant. And then I'm going to refer you to this paper, Data 2, because it was the one describing this new technology. And this afternoon, Guillermo, and well, we will give a bit more detail on the analysis based on Data 2. But basically, what is happening is this, okay? So we get our sequence, So we do the PCR, we get our reads. It's the same picture than before, okay? But what Data 2 does, does well, a removal of reads that are not correct, and also a correction of errors. Okay, so it runs an algorithm that will predict the probability that one nucleotide changes to a nucleotide by, yeah, random probability, or by mutation, or by but that it's a normal species, okay, a normal bacteria or ASB, okay. So then at the end of the day, this method, what it does is supposed to give you a better picture of reality. Okay? Clear? Okay. Yes, okay, so here I put you just to remind you how OTUs were working and how ASBs are working, okay? Well, I give also this quotation here. So ASBs are inferred by the novo process, in which biological sequences are discriminated from errors on the basis of, in part, the expectation that biological sequences are more likely to be repeatedly observed in containing. Okay? If you want to know more about this technique, please read the paper and have a closer look here. Anyway, this afternoon we will go a bit more in detail into this. A bit more information about ASBs. So this is a paper from the group of Susan Holmes, and I think it's quite nice because it's also reflecting, well, so the history of all clustering OTUs, but also ASBs. And well, what you see here, in the y-axis you have the data, or the different species, I would say, and then here you have the different technologies in the x-axis, and then is in gray you have the valid OTUs or valid ASBs, and in white the ones that are useless, so the ones that are not encoding for a real bacteria. And then, well, what you see is with the OTUs, you always get, or little capturing of the real diversity in a sample, and then a large proportion of novel sequences or sequences with errors that are not corresponding to any bacteria. This is what is happening, for example, with the novel OTU generation, or with the close reference. We also get a lot of missing OTUs. We are discarding many sequences, okay? With the SBs, you get, a full gray plot. Is that entirely true or not? Yeah, I think it's better than OTUs, but of course it has always pros and cons. Yeah. Okay. So now I have a question for you. I think we already touched upon this a bit. So, but are all the sequence identified part of the microbiome? So when you get an ASB, who has experience already working with 16S amplicon? Can you raise your hand? No? Okay. Then it's going to be difficult to answer the question. Thank you. Okay, well, I will answer the question anyway. So, well, we already touched upon this, talking about endophytes, also the endosymbiotic theory, and so on. So, well, when we sequence, we also have DNA from the host. So, if we do gut microbiome, we will get eukaryotic cells from the gut. If we do endophytes, or microbes living inside the plant, we will get contamination with plant material. So this plant material that it's amplifying with the 16S, guess what? Is chloroplast and mitochondria. And when you sequence, they are also there. So the first thing that you need to do, and Guillermo will touch upon this afternoon, the first thing that you need to do is to remove these reads from your ASV table or OTU table. That's the first thing. I don't know if you are interested in chloroplast and mitochondria, but I am not. So I would recommend to remove because, of course, the statistical methods that we are using after will be influenced by these numbers as well, and they are not part of the microbiome. Okay? Clear? Yeah. How does it look an OTU, sorry, or ASV table? Well, it looks like this. I hope you can see. I can't. But yeah, basically you have a lot of columns. Yeah. The columns are the different treatments, and then you have a list of OTUs or ASVs. It can be from 2,000 to 40,000, depending on the environment. So it will depend if your environment is more diverse or less diverse, or etc. Okay. So and then you have also the taxonomic affiliation. Okay. So you get all the OTUs, and then OTU 5, for example, here I think it's Rhizobium. Yeah. So well, and then you get the whole list for all the OTUs. Some of them will be unclassified. Some of them will be classified until family level. Some of them will be classified until genus, species, and so on. Yes? Good. Okay, so we have seen already the clustering and the generation of ASV tables. So we are going to move forward to the microbial diversity measures. Yeah? And then later on, we will move to the beta diversity and then microbiome data analysis. Okay? This afternoon, we will also do some microbial diversity measures. So we do that, correct me if I'm wrong, Guillermo. We do that with Phylosec, right? Yeah, okay. So we do that with an R package that it's called Phylosec. And I hope that you can all install it. If not, we will do it in the screen, okay? Okay, so what's the purpose of, well, this part of the presentation? Well, it's comparing the microbial communities, okay? So, yeah, so I think you could all already, you already know all of this. So how do we define microbial species? I think you are already experts on this because we have been talking already, well, 1.5 days about this. But yeah, some considerations. It's impossible to differentiate by morphology or phenotype. There is 70% of total DNA cross-hybridization between species, so that's really problematic. And then, well, I don't have to tell you, but yeah, based on OTUs, we get these arbitrary cutoffs, like, for example, yeah, 97%, as I was saying. Okay, well, and then when we are referring to, we are referring to in the single members of the microbiome, we refer them to operational taxonomic units, like OTUs or amplicon sequence variants, ASVs. Okay? Good. So what kind of diversity measures do we have? We have basically two, and they are trying to answer these questions, these two questions. So the first one is the alpha diversity, that is trying to answer what is in there and how much is there, yeah? And then that's the diversity within a sample. So if you get one plant, one replicate, that will give you a result of diversity, yeah? The second replicate will give you another measure of diversity, and then you get another plant, will be a different measure, okay? There are many different methods of diversity. I don't know all of them. I'm going to give you some examples of the ones that are most commonly used or the ones that I like more. So then, yeah, so well, here are some of the examples, like richness, that it's the observed OTUs, or evenness. Yeah, Josh already was talking today about Shannon, and Shannon basically is telling you if there is any abundant taxa in your sample, okay? Then we will move on later to the beta diversity, yeah? The beta diversity is the diversity between samples, okay? So you have 2 treatments, disease and healthy, with 4 replicates each. So we have 2 samples for beta diversity, the healthy and the disease. Okay? Okay, so here are some more details about diversity measures, alpha diversity within sample. So we have alpha diversity and species richness, that it's basically measuring how many species are present in a given sample. Okay, so every species that we have in our long list from our microbiome, yeah, will be an species, and we do the count. Yeah, so then Shannon, as I said, it's telling you about the abundances of the species, how the abundance is distributed in your samples. Then we have the phylogenetic diversity, that it's calculating the phylogenetic distance between the observed sequence. This one I like a lot. So we are gonna go a bit more in depth on this one and on this one. And there are many others, like for example, coverage, that it's estimated proportion of total diversity observed in a given data set, okay? And well, I am gonna say just this here. So I'm not very familiar with it, but I know that some people also do functional diversity. So of course you cannot do functional diversity with OTUs or ASVs. You do functional diversity with the functions of a metagenome. So when you sequence everything, so the other technology, metagenomics, yeah, shotgun metagenomics, so you can do functional diversity of genes or processes. Okay. Yes? Good. Let's continue. When talking about tools for analyzing the alpha, but also the beta diversity, yeah, we cannot, yeah, well, we have to mention QIIME2. Yeah, QIIME2 is one of the most used yeah, methods for analysis of microbiome data, and I really recommend you to use it. Another one that it's more user-friendly, it's Microbiome Analyst. Are you familiar with this one? Yes. Okay, that's nice. So you have used it before. So this one, it's quite nice. So especially when you like to do clicking instead of yeah, command line. And then here you can do different types of analysis, like marker data profiling, which will be 16S. You can also analyze metagenomics and so on, so on. There is something, yeah. So you can also analyze metabolomics. There is also another one that it's called MetaboAnalyst, and there you analyze your metabolomics data. So I really recommend you this tool. It's quite nice, and I think it's also including a lot of different normalization methods that I will touch upon in a bit. And I don't know if at the end Guillermo is gonna show something about this afternoon, Guillermo? Depending on the time, maybe we will go through it as well. Okay, so let's talk a bit about, more in detail about the alpha diversity measures. And the first method that we are gonna talk is the species count. Okay, so what do we see here? So we have plant A, B, and C, and then we have several bacteria in those plants, right? So we have Pseudomonas aeruginosa, fluorescens, Putida, and E. coli. and Pseudomonas aeruginosa, fluorescens, and then in the plant C we have aeruginosa. So anyone can tell me which plant is more diverse? Plant A, okay. And then basically plant A has 4 different species, plant B has 2, and plant C has 1. Easy, right? So then we basically can conclude that plant A is more diverse than B and B than C. Yeah? That's great. Well, this is quite widely used, and this is an example. Please have a look at this really nice paper where they look at the human gut microbiome across age, viewed across age in different geographic populations, okay? And here we have the example of US people, Amerindians and Malawans. So you can think that, well, in those 3 populations, there are different habits of, I lost the word. Lifestyle, diet, yeah, that was the word, sorry, thank you, Josh. So there are different diets, yeah? So then here in the x-axis, yeah, you have kids from 2 years old until, persons of 86. And then here we have the observed OTUs. And in blue we have US. You can see that the diversity in terms of species count is lower than in the other 2 geographic locations, okay? Good, please have a look at this paper. It's quite interesting. Then, well, let's try to look also at this example, okay? So what I'm trying to say here is that, well, species count, it's nice, but it's failing to capture the relatedness between the bacteria living in your environment, because, well, if we look here, well, we have got that in every plant we have 3 different species. But if we look a bit more in detail, I think there are different diversity measures here. So let's try to, sorry, something went wrong here. Oh, yeah. Yeah, okay. Wait a second. Yeah. No, what I wanted to say here is that indeed when we look at the species count, yeah, so, well, they are all the same because we have 3 different species here in each of the plants, but when we look a bit more detail about the diversity, okay, we see that, for example, in plant A, yeah, we have only 1 genera, that is Pseudomonas. Here we have 2 genera, and here we have 3. So then, well, observed species, indeed, is 333, is equal, but the conclusion that, yeah, A, B, and C, plant A, B, and C are equally diverse, I think I really doubt about it. So then there was a development of a new tool, yeah, to measure the diversity, and that was the phylogenetic diversity. And then, well, for example, here we have again the same samples, and in plant A we have these 3 Pseudomonas that are clustering together in this phylogenetic tree. So, well, the phylogenetic diversity of this plant will be the sum of the phylogenetic distance to the common ancestor. So basically it's 0.35, okay? When we go to the next plant, we have that the phylogenetic diversity, it's a bit bigger because we have 2 Pseudomonas and 1 E. coli, yeah? So they are also clustering here, but you know, because we have E. coli, even if it's a proteobacteria, it's a bit more farther away, so then the phylogenetic distance is a bit bigger. And then we have the plant C. So here in the plant C, we have 3 different genera. So we have Pseudomonas, Giardia, and Metanobrevibacter. Yeah? So when you do the sum of all the phylogenetic distance, you get that the phylogenetic distance is higher. So now we really, we can conclude that indeed sample C, yeah, is more diverse than sample B, and that it's even more diverse than sample A. Okay? Is it clear? Everyone? Okay. Then there are many other estimators. I think, well, if you are working with a 16S amplicon dataset, you know about Chow. So this is the formula of Chow. And basically, this is telling you also the number of species in a given sample, and it's taking into account the number of singletons, so only so in one sample, and the number of doubletons, and so on. I'm not going to go in detail, but this is another estimator of alpha diversity. Whether you choose one or the other is up to you. It depends a lot on what you want to show in your analysis. Okay, wow. I hope you can see. Well, here are some examples. For me, it's really bad. From far away is better. Okay, so here I show you some examples of diversity. So, well, it's in green we have leaf endosphere. In yellow we have root endosphere, and with the star in red, I think, you have the rhizosphere. So you can go also into this paper and look a bit more in detail about what they do. But I found it quite nice that they have several indices that we even didn't show today. So here is the observed OTUs, Shannon, Chow, ACA, ACE, evenness, and coverage. And it's quite interesting because with the 3 indices that we have seen, observed OTUs, for example, we see an increase of diversity. So here in the y-axis you have the diversity, and then in the x-axis you have the different samples. And you see, for example, that in the leaf endosphere you have lower diversity than in the root endosphere, and in the root endosphere and in the leaf endosphere you have even less diversity than in the rhizosphere. So that's why I was telling you at the beginning how important is to choose well the method that you are going to use for your microbiome analysis. So because imagine that you don't need here really deep read sequencing. In this case in the leaf or in the endosphere. So maybe in the rhizosphere you will need a bit more depth. Yeah? Good. Okay, so we are reaching the top of the complexity, I think, and now we will go down a bit. So we have seen already the microbial diversity measures, so the alpha diversity, yeah? And now we are going to start with the beta diversity, okay? You all know what is beta diversity now? I think so, yeah? Good. So, the beta diversity is trying to answer how similar or different are your samples, your treatments, okay? So if you have gut microbiome and a skin microbiome, so how different are those 2 microbiomes in terms of, general microbiome, and then also, if you have endosphere or rhizosphere, so on. So what do we do to see our beta diversity? Well, in general, there are many types. So we do principal coordinate analysis, okay? But there are many others. Constraint analysis, there are so many, and I can give you some reference to look at it. But all of them, their goal is to do a reduction of the dimensions, and then to map the distance between samples. And then this is what is happening. So here I'm showing you a PCA between of the human microbiome, okay? And then here we have the PCA2 and the PCA1, and you see a percentage of explanation in each of the axes. So what this is meaning is that the principal component 1 is explaining 30% of the variability between your samples, and then the PC2 is explaining the 4% of the variability between your samples. Then when you go into the PCA, what you see is that we have different parts of the human microbiome. So we have the intestinal microbiome. In blue, we have the oral microbiome, nasal, skin, and urogenital. And what you can see is that, well, most of the compartments or the parts of the human microbiome are different among them. So if we compare gastrointestinal with oral microbiome, they are going to have really different microbiomes, okay? Yeah? So then our next step would be to see which microbes are making the difference between those two environments, right? So for that, we are going to do differential abundance analysis. And we will do also this afternoon as well this, okay? But let's go a bit more in detail about the beta diversity, okay? So as I already mentioned, the beta diversity, it's meant to differentiate between the samples. That's clear for everybody. So then how do we measure this difference? We measure using distance metrics, okay? So imagine that we have these two microbiomes. So if I ask you, it's written there, but I'm going to ask you anyway. So here we have this microbiome, and here we have this other microbiome. So how will be the alpha diversity in the microbiome in the left? 3, right? Because we have the blue, we have the heart, and then we have the triangle, right? So then in the sample in the right, we will have 4, because we have the heart, the triangle, the purple thingy here, and then the blue. Yeah? And then the beta diversity would be 2, because we are comparing 2 samples. Clear? Okay, so I'm going to give you one example of to measure the beta diversity, and it's the Jacquard index, okay? So the Jacquard index measures the dissimilarity between samples, yeah? The thing with this one is that it doesn't count for abundance. So basically, if you have an OTU or an ASB, for this index, it's going to be a 1. If you don't have it in your sample, it's a 0. So it's a 1-0 matrix, okay? And then here I put you several microbiomes, and then we are measuring the dissimilarity, although I also put here the similarity, okay? So then here, for example, in this case, we see that the Jacquard index is 0. This one, we are sharing half, so then it's 0.5. And then here we are not sharing anything, so then it's 1, okay? So this is the basis for building this kind of plots, okay? Guillermo, did I forget something? Okay, good. Well, as I said, I am going to show you only one, but there are many, really. I don't know all of them. So here I put you a list, yeah? These are the most used ones. We have the Jacquard, the Bray-Curtis distance that is taking into account the abundance, okay? And then we have many others that are taking into account the phylogeny as well. But there are many. So if you want to know more, please visit this paper that is describing quite well all the different measures that are in there. An example, I think Josh will also touch upon this one. This was a work that we did in 2017 by Juan Perez. And then here we had several bean cultivars going from modern to wild, even land races. I'm not going to go in detail because Josh will go more in detail tomorrow, I think. Yeah. And then, well, we did a constraint analysis. It's another type of analysis, but it's similar to the PCOA. Here we basically we say, okay, I want to plot only the difference between the ancestral, modern, and land race. And this is what we see in the plot. So in green, we see the ancestral microbiomes. In red, we see the modern microbiomes. And then in blue, we see the land race microbiomes. And then in each of the axes, you get the explanation. Yeah? And today, for example, in the afternoon, Guillermo will show you how to say, okay, so I have a statistical significant difference between my two samples, ancestral and land race, okay? So basically it's to have a differential abundance, yeah? So let's move to the next step. I hope it's clear with this example. So now we are gonna move, another peak of complexity. We are gonna move up into the microbiome data analysis. Are there any questions? No? Okay. Let's move on. Okay. Well, this is a good example of metadata, actually. So, well, here we have roots, we have shoots, and then here we have again roots and shoots, and then we have plus pathogen minus pathogen, okay? So when we are talking about microbiome data analysis, we are talking about an extremely complex data frame, an extremely complex data set, okay? So don't underestimate. So there are many properties that we need to take into account. And the problem is that, well, I am a biologist, I know a bit of bioinformatics, but I'm not a statistician, okay? So you really need to know, or have someone that knows about the statistics to do all of this. But anyway, there are some things that, well, it's common sense, and I'm gonna try to explain to you, okay? So there are many properties that we need to take into account, and one of them is the sparsity, and the other one is the dynamic range. What is sparsity? It's the amount of zeros. So when we do microbiomes, when we sequence, we get really a lot of zeros. And statistical methods don't like zeros. Yeah? So that's one problem. The other one is that there is a big dynamic range. So what that means? Well, we get from 20 reads to 5,000 reads. It's really a big difference. Yeah? So we need to take that into account when we are doing the analysis. Clear? Next thing, the compositionality. Yeah, that's another thing that it's quite important. Actually, it's one of the most important things. And for that, there are many methods that have been developed in the last few years to take care of this, of the compositionality. What is the compositionality? Well, here we have plant 1 and plant 2. And we have 2 bacteria in each of them, the red one and the blue one, yeah? And then we have 8 in the plant A, in the plant 1 of the red, and 8 in the plant 1 of the blue, and well, we have 4 and 4. When you calculate the relative abundance, all of them are 50%. But this is double. So this is a real problem because we have different sequencing depths. So we also need to take into account this when we are doing our analysis. And for that, all of these methods, yeah, are taking into account these kind of, well, problems. I'm not going to go into detail here. This, we will do it in the afternoon. I mean, we will not go in detail in the afternoon neither, but I also I like to leave it here for you, for the ones that are more interested in this kind of topics. Well, this is a detail of the metagenome-seq analysis pipeline, where we try to deal with biases of high abundance taxa. And well, here is the whole pipeline. And well, this afternoon we will run it. You have these slides in case you want to go a bit more in detail, okay? Okay, so we are reaching the end of the presentation. So now it's gonna be really easy. So we have our microbiome data. We have done the beta diversity. We know that our treatments are different. We know that they have different diversities. Now, which microbes are making the difference between our treatments? Well, for that, we have also many different methods, okay? I cannot tell you which one is the best, because all of them, they have pros, and all of them, they have cons. So it will depend a lot on your question and on what you want to see in your data. Sorry. So for example, this afternoon we will use metagenome-seq, and there we will use zero-inflated Gaussian model that Guillermo will explain this afternoon. But there are many others like HR and well, DSEQ too. I don't know if you are familiar with RNA-seq, but DSEQ2 is widely used in RNA-seq data to find differential expression expressed genes. So I think this is also good to use, and many others. Okay, but at the end, this is the final result. Okay, what you want to get is a list of microbes that are well, more present in your treatments or less present, that could be performing the function that you want, okay? So, and I'm gonna explain you, well, this is an example of one of our papers. And well, here we were looking at health and disease. Josh, I think, will go more in depth tomorrow. You already saw this picture, I think. But yeah, so here you see the plants that are dying. That's the conducive soil. You will see more tomorrow. And then the suppressive soil, where plants get less sick. So basically it's disease against healthy plants, okay? So what we do with the differential abundance is to contrast these two conditions, healthy and disease, yeah? And then here it's a volcano plot representing the results. So in the in the x-axis you have the log fold chains, and in the y-axis you have the log odds. And then here in yellow I am representing the microbes that are equally represented in all the samples, in the two samples, in healthy and disease. So most likely those microbes have little to do with health and disease, okay? And then in green are the microbes that are more abundant when the plant is sick, and then in blue the microbes that are more abundant when the plant is healthy, Okay? There are many ways to visualize this. You can visualize using bar plots, box plots, different ways. We found this way and we like it. So this is what we call bubble plots. And it's representing the statistics that you see in this contrast. Okay? So in yellow, we have the microbes that are common between the healthy and the disease. Then in green, you have the ones that are more common in the disease. And then in blue, the ones that are more common in the healthy one. And for example, in this case, we found that mainly Bacteroidetes and some members of Proteobacteria were highly abundant. How do we read this plot in terms of phylogeny, of the taxonomy? Well, we go from outside, where we have the higher ranks of taxonomy. So here we have phylum, which will be, for example, Proteobacteria or Bacteroidetes. When we go to the inner circle, you get, I think it's class. Then you go inside, and then you get family, genus, and so on, so on. So you can use also this for functional analysis, when you have metagenomes, and then look at the genes that are up and down in your metagenome. Clear? Great. Okay. Then, I don't know how late it is. Yeah, okay. Well, summary. We know the toolbox already. We know how to decide what to do. Almost. We know about 16S, 18S, and ITS. Yeah? We know what is the difference between OTUs and ASBs, right? Yes. We know the difference between alpha and beta diversity, and we know how to calculate them, more or less. Yeah? And then we know how to do microbiome data analysis. It's very complex, but we know how to do it. Yeah? And we know how to pinpoint and visualize the data of the microbes that we are interested in. Right? Okay. Then I think from my side, this is what I wanted to tell you today. And I think it's the moment to look at the... yeah, questions first? Yeah. Yeah.
So can we say that way that ASBs are showing 100% similarity, whereas OTU are 97%?
Sorry, I didn't...
It's about the percent similarity rate with OTUs 97 and with ASVs 100%. That's the question.
Sorry, but yeah, what's the... So OTUs, normally we cluster them at 97.
So like with ASVs, can we say that they are showing 100% similarity sequences?
In theory, yes. Guillermo is raising the hands. He's going to help me. Yeah, Guillermo?
The OTU problem is like you need to decide threshold. And you need to put the 97%. So people like to use 97%. That is more like a species, the same species. If you use 99%, they see that it's the same strength. But ASB is not about percent. So you don't need to, let's say, a specific percent that you are going to use as cutoff. ASBs are calculated based on the data. So then it's a proper method because you don't have to deal with which of the percent is going to be good because if you are using 97%, someone will come and tell you, okay, but why are you using 97%? And most of the people won't have an answer. While ASB is data-based, so then you track the data, you track these algorithms, and these algorithms were also validated in another. So for me, the main difference is OTU, cluster, you need to keep a threshold, and that's dangerous sometimes. ASB is data, it's based on the data.
But I think the question, if I read it right, so if you have an ASV sequence which is 100% the same, it will be classified as the same ASV.
In that case, yes.
That's, I think, your question. Yeah. Good. Okay.
Yeah, then I think so. Also, you have to think ASVs suppose that it's giving you better reality of your data. That doesn't mean that OTUs are bad. I don't want to give that impression. Actually, yeah, sometimes I prefer to work with OTUs. It depends also on your data. Yeah. So, and also the question that you want to answer and how deep you want to go. That's one of the most important things.
In one of your slides, like for principal coordinate analysis, CAP 1 and
CAP1 and CAP2.
The two coordinates in the
Yeah.
CAP X and Y axis.
So it's
Yeah,
a
the
horizontal.
y-axis. Cap1, Cap.
Yeah, cap 1, cap 2.
It's
or
the
is
body
it-
type.
It's the horizontal comparison and the vertical, right?
Yeah.
Maybe you could-
Yeah, so is the, yeah, exactly. So as I explained, with the, for example, with the Jaccard distance, what you get is a distance between your samples. Sample 1, sample 2, sample 3 from one treatment, and sample 1, sample 3 from another treatment. They have a distance between them. And every dot that you see, that I didn't say maybe, so every dot that you see is one sample. Yeah?
Yeah, this one.
Oh, yeah. So
Cap1, 11.3, Cap2.
yeah, so basically this is explaining the 11% in the variation on the x-axis. Yes. Yes. It's explaining the variation. No. So the x and the y-axis are explaining here how similar are your treatments, yeah, because this is a break-Curtis similarity most likely. So how similar are your treatments? And then the x-axis is explaining the 11% of the variation, okay? That you see on the x-axis. Then you have a 2% of variation in the y-axis as well, okay?
That explains that variation.
So previous one.
Yeah, previous one.
So this come to next one. Yeah. Pre-curtaceous and unweighed unifract, weighted unifract. What
So perhaps it's asking what is the unweighted unit factor.
Yeah, those are containing phylogenetic information. So we normally use break-Curtis similarity and principal coordinate analysis. The other two are containing phylogenetic information, and yeah, could give you a different picture. I think you can look at more details into in Chime 2. You will get more information about it, and also in this paper. Yeah, so it depends what you want to represent. Yeah.
We can't predict that this is the best one, but still, which one do you prefer using? Like this will give the maximum like common.
I think. Well, that's a good question. Normally, when you run Phylosec and when you run these algorithms in Chime, you get all the diversity measures. So, and normally in papers, what you see is observe OTUs and Shannon and phylogenetic distance. Those and Xiao. So those 3, 4 are the most common ones. But also, I'm not going to be tired of repeating always the same. It will depend a lot on your question, what you want to answer. Yeah? Because every index is giving you a slightly different information. So some give you information about observed OTUs, so different OTUs, yeah, that you get in a sample. Some others give you information about abundance and phylogeny. Some others only phylogeny. So it will really depend on. Yeah, also your hypothesis. What's your hypothesis? Your question to the data. What, for example, if I'm working with endosphere and rhizosphere, and my question is what is more diverse in terms of number of OTUs? Well, I will show observed OTUs. If it's there is there any more abundant taxa in the rhizosphere than in the endosphere? Then I will show Shannon. Yeah. If I want to see the phylogeny, then phylogenetic distance. Yeah.
Because one, like, is it similar? Can we use it interchangeably? Because in one table you showed that do we represent the the ASVs and the OTUs similarly? Like you represented in a table where you showed them together.
So you mean you are asking if the ASVs, no, the
And
OTUs,
de novo OTUs, like
the novo OTUs are
ASVs, are they comparable?
the
Like,
ASVs. I don't know if there is any study about that. It would be nice to compare. Yes. Yeah. So my guess will be that a big proportion will be similar to the ASVs, but I cannot be 100% sure. I don't even know if there is any. Well, I'm sure that there should be a study comparing ASVs and OTUs and de novo OTUs. Yeah, but I didn't see. Yeah, it's a very good question. I mean, when you do a representation of your OTUs in a bar plot or whatever, you represent them in the same way. You don't do de novo and reference no, you do it in the all together. They come to the same table. When
to use OTUs and when to use ASVs? Is there any guidelines? Or we can interchangeably use it?
I think, yeah. So, I mean, there is nothing wrong with OTUs. There is nothing wrong with ASVs. So, OTUs were used in the past. Okay, I'm not saying that it's old-fashioned. No. So, when you are doing your microbiome analysis, you only have to take into account that they are clustered at 97%, 98%, whatever you choose. ASVs is a new way of analyzing your microbiome data, where it seems that it's based on all the papers that are published, that is giving a better reflection of what is happening in your microbiome. So if you want to do both, it's accepted. So you can compare, and I'm sure that you will get a lot of overlap. Of course, you will get some things that will be different, that's for sure. But in general, you will get the same picture with both types of analysis. The other thing is that with ASVs, you are really trying your best to go to species level. Yeah. So maybe for your analysis, depending on your question, that is what you need. Yeah.
Any more interactions?
There is a question.
new OTUs.
Yeah, that's a question that was so.
I think the same question was yeah.
Yeah. So it's no problem. It's good to I can repeat. I'm sure that there will be a overlap, but yeah, I don't know if there is any study doing that. I'm sure that there will be. Yeah, but yeah, I didn't look at it. We can have a look and then tell you tomorrow. Yeah, but very good question. Yeah.
Kumar, before we started with the lecture, the idea before we go to lunch, if that's according to plan, to make sure that the scripts, etc., that we need and the files that we need for this after this exercise, that everybody has them installed correctly on their computer.
We'll do that now.
Yeah, so we can do that now, and then we go to lunch, and then
Okay.
relax.
So I think with that we have concluded the two very, very important lectures. I'm sure Victor has conveyed the microbiome terminology in the simplest possible. Right? And I'm also happy that the questions are coming, very interesting questions. I would take it as a kind of feedback that the lectures went on well. It was well received by all the students.
It's a very good sign.
It's a good sign, yeah. So let's continue it. I think today's lectures are done today. Now we will go with the practicals. So I think the floor is to the team. Where now?
So I think the best would be to try to install.
Yes. Okay, so I heard that you have problems mostly with the data 2 and metagenome, right?
Go there. Go there.
Hello, check. Hello, check. Hello, check. Hello, check.
Thank you. Okay, so we're in the guideline. This is the guideline that we have uploaded in the drive. So normally the classic way to install the packages, some of them you see they have a longer like lines, while other ones are more simple and they're just like one line. That means that the ones that are longer needs to be installed from somewhere else. So what you just have to do is copy the line. This will be one package, for example, filosec. You copy this, and then you paste it in the console, so in the bottom left of the RStudio interface, and you just press enter, and it will install it. Sometimes it will also ask if you want to update some packages. So say maybe just say no. Normally it doesn't mind. So just try that. But then a lot of you told us that it doesn't work. So you remember Luisa yesterday saying about GitHub. So GitHub, this repository of packages. So then sometimes we can use another tool to install packages directly from GitHub. So for that, we have this one. So first, you will have to install one package that is DevTools. So you will have to copy this line here, install.packages and DevTools. This package, DevTools, allows you to install packages directly from GitHub, so from the original repository. So once you have it installed, well, first please try to install it, and Luisa is there, so you have also some problems. I mean, if you manage to install it through the classic way, you are all good. But if you have problems with Data 2, first try to install this package, DevTools. And if any of you have an issue, please tell, and Luisa is there, it will help you in a moment.
Yeah, so please start installing the packages, guys, and then if you have any problem, raise your hand.
Yes, and please let us know.
Yeah, we are going to be here around.
So this afternoon, maybe Victor, just one explanation. So this afternoon, because for us when we are there, to see for who it works and for who it doesn't, it's quite complicated. So Luisa came up with a beautiful idea in that if it works, you know, on the other side of your laptop, so here on this side, you basically put a green stick on. If it doesn't work, you put a red one on. And then we know with whom it works and with whom it doesn't. Yeah? Super idea, Luisa.
And try to put it in a way that it sticks out of the laptop, so I can see it from behind or from the front.
Vertically you can.
Yeah, you can also put it in there. Yeah.
So for the practice in the
Go
afternoon,
for
I
it.
will put
You know, this is for free. So, yeah, paste. It's for free, so it's nice because then you don't have to pay, even if it works. Yeah, it's not
Anyone need help?
yeah, but that's the way. So right now it's working. So you are downloading the repository, and then it will start the installation to the internet. Yeah, you see, now it's installing all the packages that are together with this data too when you are installing, right? Yeah, so it will come. I think.
Sir, after that, it will be successful.
well, many times you get some warnings, but yeah, if you so you remember yesterday.
Yes, I like that it is blue.
Yeah, this is okay. This is installing. So, but you remember yesterday that Luisa, can you remove this to the side? Yeah, move it to the side. And then you remember that here you can click and then load the package. So you are installing now which package?
Data space.
Data 2. Well, what you can do, you update this part, this terminal, yeah, and then, no, not yet, later because now it's installing, and then once you update, you look for data 2, and then you click here, yeah, you click, you check, and then you activate. If it says loading successful or library loaded or something like that, it's fine.
So that this loading here, all this checkbox, every time whenever you ask to open the app studio, every time only we do that.
Yeah, every time.
Means now we don't need to work on data too, so we don't have to check on this.
Well, some of them are automatically checked because they are programs that are needed to run on the studio, so that's fine to make basic calculations or whatever. But then when you want to activate one of your programs that you're going to use, like Data 2, then you click here.
Something clear. I see that some of you already downloaded the guideline from some days before. Please do it again because this edition we just put it like an hour ago when you came with the problem to us. So please go back to the internet version, the one that is in Google Drive, and download because it has new info.
So the file name is the file name?
It's guidelines, so it's the same file, but we just updated it with more info. So go to the Google Drive.
Look what the dates are mistake. Look, export packages, copy, paste it. When you copy it, somehow, something about your computer change some characters. So all this quote should go. You see that somehow your computer, when you copy and paste, change a lot of characters. So be careful if that happen again. And yeah, it seems that it took the quotes and put it like with a weird, like so. Now this. So now it's installing things, and it's working. Well done. So just be careful in the next case if some things get copied wrong. Yes, you see, when you see that there is a stop here sign, it thinks that it's doing things. So, yeah, just leave it. It's which one are you installing now? Which package you put it? Yeah, but you don't have to do that one. Go here. You are installing a package. So you need to copy that things that are there. Yeah, so you copy the whole thing. For example, for data 2. So go back to the document, please. No, to the guideline, sorry. You copy from the hashtag to the end of this part. Yeah. You copy it. Okay. What? But it's it. You see, it's installing things. Yeah. Package is successfully installed. So you need to go to all of that ones, take that chunk, copy it, enter, install it, and move to the next one. And if you have another error, just let me know. Yes?
I think I have installed everything already, but I just need to cross-check whether I have installed it.
When to check if you have it installed at good, you can type library, and then a parenthesis, brackets, and then the name of the package here in the console. So for example, to check if you have data 2, write library. No, sorry, write library. and then in the brackets put data2 and do enter.
Creo que DevTools debería de ir arriba.
¿Mm?
DevTools debería de ir arriba y poner para instalar, for installing in another way, y DevTools arriba, porque si no, los que van a intentar correr solo DevTools, los de MetaUnit Sec. O ponlo dos veces.
No sé, ¿qué te refieres? No entiendo bien a qué te refieres.
Si tú estás instalando, si te has instalado Data2 y no te instala MetaUnit Sec, ¿vale?
Ah, que están corriendo todo.
Claro, ahora tengo yo ahí una que ha instalado, creo, Data2, pero no le instala MetaUnit Sec. Entonces,
Y lo hace todo.
quiere hacer directamente, entonces tiene que correr el nuevo eso.
Okay, 1 minute of attention, please, everyone. Please run the things always between the hashtags. Take it in blocks between the hashtags. So this will one big block, you try to install it. If it works, you move to the next one. Don't try to copy everything because maybe this one works, but this one doesn't work, and then you reinstall everything again. And also, last thing, to check if you have a package already installed, you have to write this. For example, this line only. Library and the name of your package. Then you press enter, and if it doesn't say error or anything, you are good. That's the way to check if you have it installed, okay?
Another way to check it, sorry Guillermo,
Mm-hmm.
it's on the right side where you have all your packages, you can click the box. You can search for the package and click in the box, and then if it's uploaded correctly, then it's okay. If not, you need to reinstall. Okay?
Anyone need help? Good.
So do we have to still do again? No.
No, if you have it installed, everything works, you're good. Then you can relax now. Yes. Other people have to. Yes. Yes. Data2, it tells you sometimes to update. If you put A and enter, it means that you want to update all the packages. S is like some of them, but you need to then. So, I mean, try N, and if it does, and try it then to do library again, and if it doesn't work, try it again, but then do A.
Yeah.
Well, just do A, and it will update all the. It will update because sometimes a package installs other packages and things like that, and it needs new version. But yeah. Exactly, and it will update a lot of other packages, but that's good.
So now it's okay if I don't put A, it's okay.
Yeah, and you see, warnings are okay. Errors, no. Warning, we're okay with a warning, right? Oh, yes.
Sir, I was doing that data too.
Yeah, that is it. Just put.
But this,
Also, what?
1 minute of attention. So Guillermo said that there is an alternative way to install the program. Okay? So, and it's using DevTools. So we are going to move this up, okay? Because this is common also for metagenome sec. So if you want to install metagenome sec, well, if data to work out the installation, yeah, and metagenome sec didn't, yeah, and metagenome sec didn't, you need to install always,
Sorry.
So you always need to have installed the installation program, that is DevTools. So for you there, please install first DevTools, and then later on install metagenome sec. Okay?
Yeah.
So
And when
DevTools is a program for installation. And then you can install then later on data tools.
this line.
Yeah, this line, data 2, and then metagenome sec. Okay? We will have a chat. Yeah.
Yep. Anyone? Yeah. What?
Anything?
Need to go around. Yeah, I'll go.
Okay, where?
Lovely. Yes.
Already have data too, because
Okay, I saw that you have the same problem. I will say it, I will resolve it there, okay? Okay, one moment of attention, everyone. I see that some of you, when you copy the line from the document and you copy in the console, the quotes are changed to like an and quote thing, some weird thing. So you need to make sure when you are copying it and when you're putting in the console, it's the same text. Because some of you are getting the quotes in a weird format. For example, you. So do you see like a...
but I already have data too. So when I clicked it here, it said something detached.
Loading required errors, then you have it good. But what I was saying is like, go for example to the guideline, to the document of the guideline. Can you go, please? Yeah, you see that you copy this. You see that these quotes are not there, and it includes a weird thing. You see the quote. So be careful with that because I saw also other students that had it. So just check what you are copying is what you have in the console. Mm-hmm.
I was just checking the.
That's good. That's installed.
It's asking for installation of RCPP. So then I went it for.
No. It's a package RCPP successfully unpacked. It's okay. If you don't see an error, don't worry about anything because it is still gonna show you a lot of things. But now you put library data 2 and it say loading required package RCPP. That means that when you load this package, it's also loading this package. But it's not telling you that you don't have that package. If there is no error, word error, we're good.
So now it's fine, sir.
That's perfect. Anyone more? Yeah. Yeah?
Sir, actually, I installed the packages, but while loading,
Yeah, that's good. But that doesn't mean that doesn't have the package. It's just like extra information that is telling you that when you load the data 2, it's also loading this other package. But that's good. Yeah. Mm-hmm.
Many people are having issues because for dev tools you need R tools. And it doesn't install it automatically. You also have to go.
True, but for me, I did later and it installed it somehow when it said not.
Yeah, for her as well. Some people.
No, but then it should, for me, it show up then an installation window. That for you happen the same? Suddenly, like some error tools, and you install it like a normal software. Yeah, but that will soon happen to everyone.
But then we said yes, and now it's still saying, please.
Because you need to wait until it's installed. It takes some time because it needs to download.
But we only have one window.
Yeah.
Yeah. See, please download and install.
You know, now it's downloading, and now it should something show up that is downloading, and then like a pop-up to install it, like a normal Windows software should show up. But didn't work for you the normal way?
This is the pop-up.
Yeah, and now you say yes,
Yes.
and then you wait.
And nothing to see. Failed.
Yeah, okay, but now it should install our tools somehow. For me, it was a pop-up, and I saw that for some other people, you saw was like that, but didn't work. If not, maybe check in Google how to install our tools manually. Yeah? It's just downloading something, an executable, and run. Anyone have more questions? Mm-hmm.
Metagenome synthesis. Their tool is already installed.
Okay. Did you check,
It's coming like the error one.
for example, here is asking you for something, so we just press 1, and then it's going to install other packages that it needs, okay? Yeah, sometimes it asks you, like, if you want to update other packages that package needs. So when you say it, it gives you sometimes an option that is ASN, so just put A, and in that one it was 1, 2, 3, so one was all. So yes, but now you're good. So, anyone more? Okay. Yes. It's okay. It's okay. It's okay. Well, if you see that it's doing things, you are good. The problem is you see error or if it stops.
So everyone, please download. Don't lag behind.
Yeah, please, and raise your hand if you need help. What? Yes. Okay, it's in. You got warnings. We don't care about warnings. So, library and data 2. Error package or file it. There is no package called R6. Okay, now install it. This didn't happen to anyone, so I'm not gonna say it in loud. But it's R6. I don't know what that package is, but it seems that data 2 needed it, so we're gonna try to install it. Okay, you see, and now you do library. Oh. We need another package, moon cell. So, moon cell. Let's see. Yeah, sometimes this happens and then it tells you this package is needed, so you need to go one and by one. It's a bit annoying, but maybe now it says another package. Glue, now glue. Just try doing that until it works. Yes. Warnings are okay. We are good with warnings. Huh?
This much I installed. This I have to install now.
You check if you have the important ones, like that too. You have it. You're good. Then did you manage to install also the metagenome sec? And the phylosec too? Okay, then you're good. Don't worry about warnings. We don't care. If we have to worry about warnings, we will never end. Is it working? Okay. Yeah, for me it happened before when I tried to install it, something with our tools. I don't know why it happens sometimes. Yeah? Error. You got the same. You remember what I said? That sometimes when you copy the line, the quotes get copied as this unquote thing. So, no, just like, look, if you do up, it's the same line, but look at the line. You see that we have this, and if you go to the command line, you have it here, where you have the command line. Sorry, yes. Open it. Please. Okay. Don't you see that the quotes somehow when you copy it in the R console are changed by a lot of things? Like compare this line with the line that you have in R. You see that the quotes are put like and quote and the comma. So that's a problem. So just
Exactly, you have to copy.
exactly need to be exactly the same. So you need to go to here and you know all that parts will be just this.
Right.
So change all that
Right,
thing
I got it.
for the thing. Yeah, double check always that what you are copying in the Google document is the same that what you have in the command line. That I see that is happening in a lot of computers. Yeah, one moment. You good? No error? It's okay.
Error failed to install that. But earlier I installed this.
But if you have it installed, don't try to install it anymore. Here, when you are loading it, it says you have done package genome info db data. So try to copy that and do install package and that name. Always the package name between like quotes. So install the package and that name. And if it maybe it tells you again like that you need other package, just install the package that it tells you. See, dime, Vic?
I think she needs to update R2.
Yeah, I see that some people have that problem. But sometimes it show up an installation thing. No.
So yeah, it's R2s.
Yeah, some people have that problem.
GitHub or what?
No, the thing that one you need to install it outside and it's different. Es una movida. Es una movida.
Okay.
Yes.
So what do we do with those?
If you put in Google tutorial install R tools, because she had to do the same. You need to download like an executable and.
So you know how to do it?
So the div tools has to be installed.
Then
It's not necessary. If you have all the other dev tools, it's only if you couldn't install that too of any package. But you managed to install it,
Yes, sir.
then you are good. If you put like of all the packages that are library except dev tools, you're good. Exactly. Only errors. Only errors is the problem. Errors is the problem. Yeah. Qué calor. Yes.
I installed DevTools and then I did that for I followed the command for the metagenome set. Then I did library metagenome.
You're good. If it doesn't show, you're perfect. Only errors. Yes. What? Yeah, one moment, I'm here.
She doesn't have metagenome.
Metagenome seek I'm not getting in the packages.
This is the new one she downloaded just now.
Can you try to type, well, I mean, now don't write anything because you have a stop here.
Okay.
So that means that it's doing things. It's the R tools. Because I see a lot of people with the R tools problem. Well, 3 or 4. Yeah? Is this? Okay, look. Now it's downloading R tools. So it is installing this thing. So then something will pop up and it will be like installing something, but like when you install it in Windows, that you have the thing and then you say, yeah, continue, install, blah, blah. Do that and then try, is this yours or mine?
So after that I will again, should I?
Yeah, when you see that all of these finish, something should pop up and then you should install that and then you try it again, okay? Yeah? Do you see an error there?
No.
Then you are good.
It's okay.
Yeah. If you don't see error, you are good. If you put library, it takes some package. It takes too long, especially metagenome sec and the phylo-sec take long. So I think most of people is there. Yeah. I mean, some people is having the R tool problem, but yeah. Remember to check library and the name of the package to see if installed. If nothing show up, you good. Only worry if an error show up. We don't care about warnings, okay? Yes.
So with these three packages failed install. With my email, it should run detailed by our failed install.
You can just like copy that line.
No, I've tried installing it, but it is showing that
The launch.
the download is failed.
Okay, but if you do library and the name of the package, it works.
It is like showing that there is no package.
Okay, go down. You see, but you are not connected to internet. You need to be connected to internet because it needs to download things. And check that all of you are connected to internet because it's needed. All good here? Perfect. Oh, yes. One moment.
Warning is fine.
Yeah, we are good with warnings. Yes.
So some permission denied, is it occurring or not?
Permission denied. That's weird. Which command did you run? Let me see.
So data 2 is not coming.
Did you check a library data too? You don't have it there, right? Permission denied. But this is a problem of your computer, you know?
Sir, if BIOS manager is there, then data 2 cannot be downloaded or what?
Yeah, but this has to do with some permissions inside your computer.
Okay.
Because it's kind of telling you that you don't have permission to install that in your computer.
Can we give a break? Let's go for lunch. Is that fine?
So
No?
we'll go for lunch. I can help you to see if we can fix that because that's really specific problem. But did you manage to install all the other ones? Okay. Still you're... Okay, I mean, it's not that a big deal because everything is pre-run, so you will still could be able to do the practical, but then I will help you because...
So data 2 is not downloaded, it is in between. So
No, but this, you already installed it. If you already installed it, don't try to install it again. If you have it. If you see it, first try library and the name of the package. And if it's good, don't try to install it.
Sir, metagenome set is not installed. And now here I am in between of installing data 2. So if it is not happening, so permission
Move
denied.
to the next one.
No problem.
Yeah, I mean, it will be good. It will be good. There will be just one little step that maybe you cannot generate a lot.